This meeting is being recorded. Ahí está. Okay.
Francisca is here also. Can you add as panelist, Victor,
Okay.
please?
Let me see.
Well, Francisca is going to talk just after the coffee break. So
After the coffee break.
But you can add as panelist now.
Yes, yes.
Hi, Aresu. Good morning. You are mute.
You are mute.
Hi, how are you doing?
Hi, how are you?
Fine, thank you.
Nice
So,
to meet you.
Yeah.
Okay. Who is there?
Can you hear me well?
Perfectly, Aresu.
Sure.
Do you want to try to share your screen to see if it's working?
Sure. Let me just go to the first slide and make sure.
Francisco, the
Yes.
next speaker is Francisca Guzman, okay?
It's Ruben, no?
Ruben.
But yeah, you can add Francisca as well.
Okay.
Can you see my screen?
It's black now. No, yes.
Yes.
Perfectly.
Okay, great.
Oh, beautiful. Okay. Who is next? Okay.
So when you said that it's okay to start, Victor, we start, okay? Because it's now 9 a.m.
Okay, let me see. I think 5 minutes in order to wait the other attendants and participants, please.
Okay. Victor Ruben is already there as attendee, I think.
Let me see, Ruben. I didn't see. Ah, okay.
Juan Ruben.
Yes, Juan Ruben. Okay.
And it seems that Francisca is offline now, right? Ah, no, Francisca is there also.
Okay.
Hi, everyone.
Hi, Ruben. Good morning.
How are you?
Good morning.
Morning.
So, Paco, I think it's time to
Start?
To start.
Okay. So, good morning, everybody. Let's start with the second day of our Congress, of the International Congress on Science and Technology of Complex Fluids. In this edition and this special day, let's start with Professor Aresu Arbekani from Purdue University. She will talk about elasticity induced instabilities, flow through porous. So when you want to start, Aresu, please go ahead. Thank you.
Sure. Thanks for the introduction. I'm going to talk about elastic induced instabilities and flow through porous media. Also, we are going to talk about multi-instability in array of pores and pore network, as well as ordered and disordered porous media, and having some discussion about flow around cylinders. So the reason we are interested in this problem is that it has applications in targeted drug delivery as well as flows through cilia that are covering airways and lungs, where they can lead to trapping of particles and their mucus that are covering these ciliary arrays. Also, it has applications in biofilms that can form and lead to infection for human and animals, and they form everywhere from prosthetic devices to catheters that are used to transport body fluids. It has also applications in enhanced oil recovery, where polymers or surfactants are used for enhanced oil recovery, because primary recovery is only recovering about 20% of the oil, then water flooding will lead to additional 15% recovery. So in order to be able to get access to additional oil or hydrocarbons in the subsurface reservoirs, either surfactants or polymers are injected, or steam and other microbial injection is also a new technique being used. But for some of these, polymers are relevant, and it's interesting to know how elastic instability can play a role. For groundwater remediation as well as filtration, we have water and fluid going through porous membranes or through subsurface reservoirs, where these are examples of porous media. Examples of elastic instability, elastic induced vortex have been studied in some canonical geometries like constriction flows or flow around cylinders. And I'll review some of those before getting to what we've been doing on flow through porous media. So the flow through constriction and the vorticities that form had been studies by Lanzaro and Yuan, and they looked at the phase diagram, looking at the range of contraction ratio as well of Weisenberg number, where Weisenberg number itself is the ratio of elastic forces to viscous forces. And they observed that as this contraction ratio increases or Weisenberg number increases, these upstream vortices form in these constriction. And as Weisenberg number increases, these upstream vortices can become even asymmetric or transient flow occurs for these geometries. Also, if you add inertial to this flow, inertial effect, which is shown here in this phase diagram in terms of Reynolds number, which is the ratio of viscous force, which is the ratio of inertial force to viscous force, as inertia increases, these instability is suppressed. So the upstream vortices can go away. On the other hand, in this phase diagram, if you increase Weisenberg number and elastic forces are increased, then these vorticities get and vertical flow gets stronger, and transient flow and chaotic flow can be observed as well. For flow around cylinders, for very small Weisenberg number, the flow is symmetric and no eddies occur, but as Weisenberg number increases, these eddies form upstream of flow, and the eddies can become asymmetric, and transient flow occurs for even larger Weisenberg number. And this had been studied by a number of researchers. Another canonical flow that had been studied in the context of elastic instability has been cross-slot channels, where the flow is symmetric for small Weisenberg number, but it becomes asymmetric and elastic instability occurs for large enough Weisenberg number. And this problem had been studied both computationally and experimentally. And for these cross-slot geometries, because of the presence of boundaries, the extensional flow is not, flow field is not uniform. So to create a homogeneous elongational flow field, another design of this problem had been studied, where you can basically, by changing the boundary of this device or microfluidic device, you can actually generate homogeneous elongational flow. And in this case, the elastic instability is characterized by time-dependent motion of these stagnation point in such a flow. For two cylinders that are in a streamwise direction, for a small Weisenberg number, no eddies occurs, the flow is symmetric. For large Weisenberg number, the vertical eddies are observed between cylinders. And for array of pores, these biostability is reported, where you get these vortices in the top or bottom of the pore network. So what I'm going to talk about is starting with this array of pore network and then building into ordered and disordered porous media to understand elastic instability and the nature of flow in more detail. So we studied this problem computationally by solving continuity equation as well as the momentum equation, where in the momentum equation we have the stress, which is basically has contribution from both solvent, which follows a Newtonian constant equation, as well as polymer, where the polymer stress itself is modeled here using Finney-P constant equation. So we have stress relaxation, we have viscous stress and polymer stress that is modeled by this constant equation. So rather than looking at all the chains in the polymer network, which won't be computationally affordable in continuum approach, we model it as a spring dumbbell. So entire polymer is modeled as this spring dumbbell, and the finite extensibility of this polymer is also modeled through this function of a stress, which has three parameters basically here. One is finite extensibility length, which itself is related to maximum allowable length of polymer chain over the equilibrium chain length, as well as it's related to relaxation time and polymer stress. And the parameter A here itself is related to the finite extensibility length. So we have three basically free parameters here, which defines this constant equation for polymer stress. If you look at the range of dimensionless parameters that are relevant in this problem, we have Reynolds number, which is the ratio of inertial force to viscous force. That's very small, often for applications that I talked about, from biomedical application to subsurface reservoir application. They all have very small Reynolds number where inertial effects can be neglected. Weisenberg number, which is the ratio of elastic force to viscous force, can be large. And consequently, elasticity number, which is the ratio of Weisenberg number and Reynolds number, can be very large because of these parameters that we discussed. We here also define advective Deborah number, which is relaxation time over the time scale corresponding to advection of these polymer molecules. So if we define this advection basically between two adjacent pores. So if we have just one pore throat, then this length scale basically becomes infinity, the distance between the adjacent pores, so that becomes 0. But if we have two neighboring pores, then based on that we can calculate the Deborah number. And as we go to array of pore networks and reduce the distance between these pore throats, we get larger advection Deborah number. So now let's just start with single pore throat. If you look at the flow through this geometry, we see upstream vortices occur, and also we see stretching of polymer that is very large, close to the surface of this pore throat, and we get also these polymer stress in the form of waves advecting downstream of the throat. And we quantify the length of this eddy by looking at where basically velocity becomes 0, and that encompass all the eddies that form upstream of the geometry, the cylinder, and we use that to quantify the instability in this problem. So for example, here, if we are looking at the length of eddy as a function of time, we see these fluctuations that occur. And as we increase Weisenberg number, we see larger and larger fluctuations. Here we plot power spectral density, which shows even larger fluctuations that occurs for larger Weisenberg numbers. This is better represented in a plot of basically PDF of these eddy size as a function of Weisenberg number. And we see that initially for a smaller Weisenberg number, we have a very we have just single eddies with a given length, but as we increase Weisenberg number, we have distribution of these eddy length because the problem becomes transient, and we see eddy length basically fluctuating in time. If you look at the average value of the eddy size as well as the standard deviation, it has two slopes in the for smaller Weisenberg numbers versus larger Weisenberg number, and we use that to define a critical Weisenberg number for this problem. Now that we kind of established how we can study such a problem, we can add to the complexity and looking at two neighboring pores, where similarly we look at fluctuations of eddy length and we see fluctuations increases as we increase Weisenberg number, and the average of this eddy length is slightly larger for the upstream throat compared to downstream throat. But many of the characteristics that we saw for single pore throat is observed here, and the reason we see a smaller eddies behind the rear throat is because the polymer gets stretched as it goes through the first pore throat, but it doesn't have, as the pores get closer and closer to each other, it doesn't have time to get fully relaxed, and then it enters the second throat. This plot is basically showing similarly compared to single pore throat, where we get a eddy size of with a very narrow PDF for a small Weisenberg number, but the spread of that PDF expands as we go to larger Weisenberg numbers. Moving forward, we look at now array of pore networks, and here you see for a small Weisenberg number, we get stable flow. We get vortices that appear in the top portion of the throat, as well as the pore, and as well as the bottom portion of it, but it's quite stable. And the polymer stretching, basically the pattern is correlated to these flow separations or eddies that form, where polymer stretching occurs or is strong adjacent to the wall of the cylinders, but in the top portion or bottom portion of the throat, basically where we get these streaks of high polymer is where we have flow separation. As we increase Weisenberg number, we get the multi-stability that happens in such a problem. If you look at a particular throat, we see at some instance of time, we have no eddies that form. Some instance of time, we have eddies forming in top portion or bottom portion, or both basically areas will have eddies covering them. This is more clear if you look at the snapshots where all the different states are shown here. A pore without any eddies, two eddies forming in top and bottom portion, one eddy forming in top or bottom portion. And if you look at the polymer stress, also very well represents such a flow pattern. So for example, when we don't have any eddies forming, the polymer stress is very large adjacent to the walls. When we have the separation occurring for both top and bottom eddies, we see these streaks of polymer stress, which corresponds to the region separating the two flow state, and the flow crossing these regions of high polymer stress is a small. We look at the space-time plot for a trace of polymer stress, and for the case when we have a smaller Weisenberg number and these stable eddies form, we see these streaks of high polymer stress, which is stable as we look at the particular pore along this red line as time passes. But the behavior is very different when we increase Weisenberg number. For state 1, basically where we have two eddies forming, this is where the streaks of high polymer stress occurs. For state 2, you see one eddies, and that's where the streaks of high polymer stress is separated from the wall. For state 3, they're attached to the wall, and in state 4, we have vortices forming both states, and again, they are separated from the walls. To understand the flow pattern and how they are correlated for neighboring pores, we define this parameter, we call it F, which is basically looking at how the area of the eddy for neighboring pores here shown with I and J are related to each other. So this parameter F would be 1 if pores have similar flow patterns, and would be -1 if one pore is eddy free, the other one has eddies. So we see fluctuations going from -5 to 1, and this is more represented or more clear when we look at contours of time average of this correlation parameter. And of course, if you look at autocorrelation, the value would be 1, but and for neighboring pores that are close to each other, they are more correlated. As we move further away, they become less correlated, but there are some outlier here where, for example, 8 pores away from each other, we see actually high correlation. We don't know why that happens, but this is basically the behavior of these flow patterns. And if we average it not only on time, but also across different pores, we see that this parameters is reduced as we increase Weisenberg number, and also is reduced as we go, increase the separation between pores. As we increase Weisenberg number, we get more chaotic flow and basically transition between these different flow states, and that's why we get a reduction in this parameter. In terms of the PDF of size of eddy, we get a more narrow PDF as we look at the smaller Weisenberg number and broader PDF as we increase Weisenberg number. And that's where basically we define our critical Weisenberg number, where the slope of mean or standard deviation of these eddies basically changes. And we use that to define our critical Weisenberg number. Pressure drop is also interesting in such a problem. After initial transient, it gets to quasi-steady behavior, but the pressure drop is larger when we look at 10-pore network, and that's because we have higher flow resistance, but it decreases as we increase Weisenberg number. And the reason is that if you look at the time history of these flow patterns, there are states where there is no eddy, and then the cross-sectional area increases. So consequently, the resistance of flow reduces, and that's why we get reduction in pressure drop as we increase Weisenberg number. There are also states where we see these eddies forming in the middle of the channel, and this is different from the other states that I previously described. In the very beginning, we talked about basically this viscoelastic instability can also be useful in recovery of trapped oil. So if you think about having trapped oil in these pore throats, because of transition from one flow state to the other, that leads to release of these trapped oils from these pore space. And this had been experimentally observed for having oil trapped in microfluidic devices and then injecting that with different polymers, and the oil saturation is reduced as polymers are being used in such geometry. And one of the reasons for such a behavior can be viscoelastic instability that we talked about. Next, I want to talk about different geometry. Still, a problem is looking at flow pattern as we have polymers going through two cylinders that are in a streamwise direction, and understand how flow pattern changes for different Weisenberg number. We know for single cylinder, the flow can get asymmetric, and also these eddies can form upstream of single cylinder. So we want to know how the behavior is different when we have these two cylinders in streamwise direction. So here's the flow pattern for small Weisenberg number where there is no eddy forming, but as we increase Weisenberg number to 1.88, we see these eddies that are forming between the two cylinders. And for even larger Weisenberg number, the flow becomes asymmetric and there is no eddies. So we have basically tri-stability behavior for such a problem. And we define two critical Weisenberg numbers as we transition from symmetric flow to generation of these vortices to asymmetric flows. The parameter that we can use to understand this elastic instability is called Pactel-McKinley parameter, which had been studied by McKinley, but also linear instability problem for flow around curved streamline had been studied by Shakfi and co-workers. Where in this Pactel-McKinley parameters, basically parameters that show up are tensile stress along the streamline, the shear rate, radius of curvature of the streamline, and velocity field. So because we have computational solution of this problem, we can calculate each of these parameters and see what happens and when we get these M going above a threshold leading to instability. So let's look at each of these terms. This is the tensile stress along the streamline. So we have large values occurring in the wake of the bottom cylinder. For radius of curvature, it has a very complex pattern, but for velocity, we have it large close to the side of the cylinders, and shear rate is also large close to the side of the channel, near the wall of the channel as well. But if you look at this ratio, we see basically for a smaller Weisenberg number where we don't have any eddies, we see these maximum M occurring on the side of the rear cylinder, and we have eddies in between cylinders, we see maximum occurring also in between the two cylinders. So we basically looked at this maximum of M parameter I've discussed for a different Weisenberg number, and we see the location below a threshold is side of the rear cylinder, but above a threshold, it's in between cylinders. And that's actually the critical Weisenberg number we found. Also, we see the change in a state or flow pattern as we increase Weisenberg number. Looking at the PDF of eddy size, it clearly shows the tri-stability. Basically, for small Weisenberg number, there is no eddy. For very large Weisenberg number, also there is no eddy. But in the intermediate region, we have PDF of these eddies that form between two cylinders, and the power spectral density shows that the amplitude also increases as we increase Weisenberg number. This is a plot of eddy size as a function of Weisenberg number, where the green line is showing first critical Weisenberg number, which here it shows that it's independent of the finite extensibility of the polymer, but the second critical Weisenberg number is dependent on both these finite extensibility as well as the viscosity ratio. Beta here is the solvent viscosity over total viscosity. And as we increase solvent viscosity, the flow becomes more stable. As we reduce the L parameter, which is the extensibility of the polymer, also becomes more stable. We quantify asymmetry in the flow by looking at the flow rate that goes top portion of the channel versus bottom portion of the channel. And that shows basically the flow becomes asymmetric close to second critical Weisenberg number. And it also depends on the finite extensibility parameter. And the asymmetry across the upstream cylinder and downstream cylinder also shows how the flow changes as we go travel through the channel. Basically what we see is that eddies between cylinders can become also have a slight asymmetry even before we transition to the third flow pattern after crossing basically the second critical Weisenberg number we discussed. If you look at larger distances between cylinders, we see that the formation of eddies can get even suppressed when we go to distance that is 3 times diameter of these cylinders. So the critical Weisenberg number, then the state of flow depends on both separation between cylinders, but also confinement has an effect on these problems. So for example, here we're looking at the second critical Weisenberg number that is different as we increase the distance between cylinders. It goes away because you see the black line that is the eddy size is very small or non-existent. But also the confinement here has an effect. So as we go to the smaller blockage ratio, the blue line has larger critical Weisenberg number. Another interesting observation that we have for such a problem was that we saw that if we increase Weisenberg number and decrease it the same way, we see flow hysteresis. So here we're looking at the eddy size as we increase Weisenberg number and then we decrease it. So the flow behaves differently. And because of these streaks of high polymer stress, this flow has basically memory for finite time. So that's why we see such a hysteresis that is observed when we look at eddy size or also this asymmetry parameter that we defined earlier. We did collaboration with Jeff Costa and Derek Walkama at Tufts University to look at elastic instability in 2D porous media. We did computational analysis of such a problem, and they built microfluidic devices with the same distribution of these cylinders, and I'll share those results here. What I'm showing here are from computations. So Manish Kumar, my PhD student, was working on these computational results. So you see for this is Weisenberg number 0, Newtonian fluid, and the flow is stable. As we increase Weisenberg number to Weisenberg number 5 for these ordered geometry, we see these regions of high velocity, and we see elastic instability that occurs. So this is highlighting some of these regions of high velocity and velocity fluctuations as well. So if you look at the same Weisenberg number 5 for ordered geometry, this is the flow state you already saw, but this is also polymeric stress that shows these streaks of large polymeric stress that form in such a geometry. And in regions where we have high velocities, we have also high polymer stress. Now, if we look at disordered geometry for Weisenberg number 1, we also have some regions of high velocity and velocity fluctuations. It also exists for larger Weisenberg number 5. And we see these regions where we have some preferential path for flow to go through these porous media. And this preferential path is basically caused by these regions of high polymer stress, where it's generating basically these long tails of polymer stress is creating these preferential path. And for this particular geometry that we talked about, for order geometry that we talked about, as disorder in the system increases, disorder can even suppress viscoelastic instability. So what we wanted to study also is transport through these disordered and ordered media. So on the left, you're looking at the results from experiments, and on the right from Derek and Jeff's experiments. And on the right, you're seeing our computational results for the same problem and same geometry. So here, as we go to larger Weisenberg numbers for these geometries, we have basically flow instability. And when we get also disordered experiments, we have flow instability, and there are regions where we have these large velocity and large velocity fluctuations as well. Now we basically both computationally and experimentally injected large number of particles and looked at dispersion of these particles. So here you're looking at the color is basically showing time. And so the color becomes more purple color as time passes. But this is showing trajectory of each of those particles. So the spread is very small in ordered media. And as we go to, for smaller Weisenberg number, as we go to larger Weisenberg number, because of the elastic instability, you see larger lateral dispersion of the particles and also slightly larger longitudinal dispersion of particles. This is also clear when we look at the PDF of these particle position, and this is basically the sharp peak is corresponding to this, the blue line is corresponding to case of ordered geometry for small Weisenberg number. As we go to disordered geometry, but also ordered geometry, larger Weisenberg number, because of these larger fluctuations in the geometry, we have more spreading of these PDF. This is looking at transverse dispersion. Looking at longitudinal dispersion, and we see that most of them are similar, but the one for disordered geometry and larger Weisenberg number, it has less longitudinal dispersion. And this is consistent with saying that in disordered geometry, fluid elasticity can suppress the fluctuations in the system. For the next few minutes, I want to switch gears and talk about another topic still related to elastic flow and how velocity fluctuations evolve, but it's not related to flow porous media anymore. It's related to collective motion of microorganisms. So we know if you're dealing with bacteria, they have velocity fluctuation in the system. We get regions of high concentration and low concentration. And if you look at, for example, bacteria either on agar plate, which is the one on the left, or the one in between thin film, similar behavior is observed. And here you're looking at contours of vorticity, where you see also these patches of large vorticity and velocity fluctuations in these suspensions of bacteria that are swimming around. So what we were interested in is to understand how fluid elasticity modifies these velocity fluctuations. And the reason we are interested in such a problem is that it's applicable to biofilm formation, when biofilm itself can be can show complex flow behavior, but also thin film in the ocean, where mucus of different organisms can be trapped in the thin layers, and also large concentration of marine organisms are present. And biofilm itself, which I already talked about, which can happen in around prosthetic devices or catheters using body fluids, and can lead to infections. So if you look at extracellular polymeric matrix for biofilm of Pseudomonas aeruginosa, for example, it shows not only shear thinning, but also a large normal stress difference, and the normal stress difference itself has a shear thinning behavior. So there are polymers present there that their concentration can be large, 1 gram per liter, or their molecular weight can be large as well. So that leads to relaxation time of 1 to 100 seconds. Also, here is the numerical simulation of the system. We are looking at microorganisms that are modeled here as rods, self-propelled rods. Here we are looking at pushers where thrust is generated behind the body, and we see these velocity fluctuations in the system for both Newtonian fluid, there were a number of 0, but also for viscoelastic fluid. And viscoelasticity basically suppresses these velocity fluctuations. So this is quite opposite of basically what we discussed when we were looking at flow through porous media. Here, we were looking at pushers. Now, if we look at pullers, we see that velocity fluctuations exist in suspensions of pullers as well. Fluid elasticity doesn't change those much, but clustering or aggregation formation for these pullers are very different from pushers. And this is more clear when we look at them side by side. The one on the left is for pushers and the one on the right for pullers. We're looking at contours of polymer stress, and they are stretched behind the swimmers. But you see these asterisks that are forming for suspension of pullers, but these rods that are, or these swimmers are parallel or anti-parallel for the case of pusher. This is more clear when we look at probability per correlation function, which has peaks around 0 and pi. And for the case of pusher and for the case of puller, it has large values for different angles. So that's because they form these asterisks. If you quantify cluster formation by looking at probability per correlation function, we see as a function of R, we see peaks both in Newtonian fluid, but for viscoelastic fluid, for pushers, these peaks are larger. And so we see more clustering for viscoelastic fluids for suspension of pushers. And looking at velocity correlations, fluid elasticity reduces it for pushers. But where we have these velocity correlation go to 0, which tells us about size of these patches of large velocity fluctuations or clusters of form, is still unchanged by the presence of polymers. This is the enhanced clustering that occurs for viscoelastic fluid is consistent with experiments that are done for sperm, where suspension of sperm are observed in a standard medium, more viscous medium, and viscoelastic medium, and more clustering was reported for viscoelastic medium. Because we have access to the computational flow field everywhere in the domain, we can look at energy balance, where we have kinetic energy balancing elastic energy, the energy input at the swimmer, and part of it is being dissipated by viscous dissipation and dissipation because of presence of polymer. We can calculate each of these terms and see how it evolves. We also look at local alignment parameter to see how these neighboring swimmers are aligned with each other. And looking at fluctuations of kinetic energy, we have the time scale for these fluctuations that is on the order of convection time scale, size of swimmer over its swimming velocity. We see that there is no correlation between the size, RC here is where velocity correlation goes to 0, which is size of those patches of large velocity fluctuations, and alignment parameter we talked about. So the alignment itself is a local clustering that forms. When we look at polymeric stress, we see kinetic energy and elastic energy are anti-correlated, and the time scale of fluctuations reduces to relaxation time of the polymer. Kinetic energy spectrum is shown here, where we see the slope of -8/3 for wave numbers, for smaller wave numbers. And this is consistent with experiments for suspension of E. coli and Bacillus subtilis. And here you may say this number is different, but they plotted basically k-square wave number squared times kinetic energy, so they are the same. Also, you may say this is for smaller wave number, these are for larger wave number, but that there's a difference between experiments and computations. Computationally, they are still the same range of -0.1 to 1 dimensionless wave number. And computationally, we can access to much smaller wave length, but experimentally you cannot basically resolve flow field smaller than bacteriocytes. So that's why that characteristics of these plots look different. But what we see is that we add polymers to the system. Suspension of pushers have a smaller kinetic energy at a smaller wavelength, but larger kinetic energy for larger wavelength. So basically, we have a mechanism for transfer of energy from larger eddies to smaller eddies because of presence of polymers, which is absent in the absence of polymers, because if you look at collective motion of these bacteria in Newtonian fluid, these are small Reynolds number, and viscous dissipation occurs at every length of scale, basically. Let me see how I'm doing on time. So for the next 2-3 minutes, I will just highlight some of the other works we do in my group very briefly. So we look at microbes and cells in flow and how flow itself leads to trapping of microbes. We look at how fluid elasticity in vertical flow can lead to emergence of limit cycle and aggregation. We've looked at the sperm in shear flow and how basically lateral migration in Poisson flow, for example, occurs for sperms. And we looked at buckling of the sperms in extensional flow. For example, if you have extensional flow and you look at motion of a sperm, you would see that it actually buckles at large enough sperm number, which itself is the ratio of viscous force to elastic force. This is also collaboration with Jeff and Derek, where they did the experiments, and my students, Manish, performed the computations for this problem. And you see computationally, we can capture exact same behavior as is observed in experiments. And if you look at the space-time plot for these, for radius of curvature, basically as function of arc length and time, we see these large curvatures that occurs in experiments. We have single buckling mode for small sperm number. For large sperm number, we see multiple buckling points, and we see exact same behavior in computations for using the same sperm number. As we increase the sperm number, we get more buckling instability and multiple modes of instability. We looked at microbes in complex fluid, not only from their swimming speed, but also how they interact with surfaces and interfaces, and how shear thinning properties and viscoelastic properties of the fluid affects their attraction to surfaces. Fluid elasticity brings basically these undulatory swimmer close to the surface, but it doesn't move as fast because of its head being attracted strongly to the surface. But if you include shear thinning, it goes much rapidly, because this shear thinning leads to generating corridors of small viscosity medium around the swimmer, and that effective confinement leads to faster swimming. And we moved it also to more applied area, looking at its application and how we can basically detect microbial contamination in pharmaceutical settings. So for that, we use optical sensing or Raman spectra, and we couple it with machine learning tools to basically be able to detect microbial contaminations. So here is confusion matrix from the machine learning tools we developed, where we have basically the different strains of bacteria, fungi, cells that are present in the system. And then we use optical sensing and are able to detect and predict the bacterial contamination with very high accuracy. So you see, basically we have accuracy above 97%. So with that, I'm going to conclude. In the first part of the talk, I mentioned unstable eddies that appear upstream of single pore throat, and I talked about how large polymer stresses leads to multi-instability inside pores of converging diverging channels. We talked about eddy formation in the case of a streamwise direction cylinders in the flow, and we talked about how the streaks of high polymer stress guide flow in porous media, and looked at dispersion in porous media. And in the second part of the talk, I talked about fluid elasticity and how that suppresses velocity fluctuations and induced mixing in suspension of pushers, and how it increases clustering in those suspensions, and it enhances wall clusterings as well. And with that, please let me acknowledge actually the role of those students who participated in what I presented today. Most of what I presented today, the work on porous media was done by Manish Kumar, as well as the work on sperm that I very briefly mentioned. And the work that I mentioned on elastic instability or elastic stresses in suspension of microswimmers had been done by Gao Junli, who was a student in my group and very recently joined Shanghai Jiaotang University as an assistant professor. And with that, please let me know if there is any question.
Thank you, Aresu, for a really interesting talk. Let's open the session for questions. Juan Ruben is raised his hand. Yes, Ruben?
Yes, I would like to ask a question.
Yes.
Thank you, Aresu, for your wonderful talk. In the second part of your talk, you presented these simulations on pushers and pullers in viscoelastic media. Are they, do they move in 3D or in 2D, or what are the conditions? Because you also presented some comparison with the swimming of sperm cells. So what are the details?
The comparison is very qualitative, not quantitative in any way. The experiment was, of course, a sperm in near substrate, swimming near substrate, but in our case was 2D problem. So you can think of basically 2D movement of some bacteria in a thin film.
All right, thank you.
Is there another question from the audience? Let me see. Yes, Juan Dueñas, please let me open the microphone. Maybe Victor, you can open the microphone of Juan Dueñas, right?
Yes, yes.
Please, Juan, go ahead.
Thank you. Well, and thanks for your very interesting talk and informative talk also. I was wondering if you said your study the motion or the flow stability under different geometrical conditions. One was an order arrange of pores, I guess you could say, and the other one was a disordered geometrical arrange of these pores. And I was wondering if the problem that your team or you solved was a 3D or a 2D problem, and also if this if you found an expression for the velocity field in relation to the different arrangements of or geometrical arrangement in this case.
So we did find velocity field for these different arrangements that I talked about. In terms of 2D versus 3D, some were 2D and some were 3D. For example, for everything that I talked about, flow through porous media or the two cylinders flow around two cylinders, they were 2D, but we have done 3D flows as well. They are computationally more expensive, so it's slower for us to get the results, but we are in the process of getting those results for 3D geometries as well. For the case of swimmers, the one on suspension of rods that I showed was two-dimensional calculation, but we have done also a three-dimensional calculation of swimmers between two walls using resolved computational methods. We have also done some semi-analytical methods where we look at we model bacteria as point for singularities, and for those the flow field and patchiness and geometries you get are also 3D. Another study which I didn't talk about here, but we looked at basically microbes interacting with droplets and how that creates patchiness in their distribution. Those are all 3D as well.
Okay. Thanks. Thank you very much.
Thank you for the question.
Yes, Francisca, go ahead.
Hi. Thanks, Areso, for your talk. Very interesting and fun. So my question it's in the beginning of your talk, you told us about your inspiration behind the problem. So the problem is try to transport maybe cargo delivery or micro swimmers through porous media in presence of a viscosity term, let's say. And my question is, after all your work, what would you recommend if you want to transport, let's say, microparticles through a porous media? Will you choose like the disorder scheme, maybe, or the order one, or will you transport your particles through a throat? What can you say about that?
Mm-hmm. In terms of geometry, I would say most of the time you have limitation on the geometry, so you can't really change the geometry. And if you think about the application of, for example, either tissue or subsurface reservoirs, most of them are disordered media. But it helps us to understand whether in which of these geometries, for example, elastic instability is suppressed or increased, and how that can be used, let's say, for application of enhanced oil recovery. When you want to get more oil out, you want to get more elastic instability. So you want to basically work in a parameter space where you can get more elastic instability. Though I should mention that, for example, polymer injection for enhanced oil recovery, elastic instability is not the only mechanism. Another advantage of injecting polymers is that it increases viscosity, and that suppresses viscous fingering instability. So I guess there's a trade-off. You want to, on one hand, suppress viscous fingering instability, because then leads to, let's say, when you're injecting the polymeric fluid, if a viscous fingering is present, then there will be some channels when fluid is going to travel faster and doesn't get any oil release. But on the other hand, having some elastic instability would help with getting some of the trapped oil ganglion to be released. So I guess it's not easy to make a recommendation, because it very much depends on the geometry of the porous media as well.
Mm-hmm. Thank you very much.
Thank you.
Thank you. Is there any further question for Aresu from the audience? Okay, so if may I have a couple of questions, Aresu, if may, when you talk about this flow in different geometries, in both random and periodic, it's quite interesting to see that the flow from the random distribution is higher than in the case of periodic distribution, right? So I don't know if you checked the case of other periodic distribution, meaning that instead of have this like a square lattice, to have a different ordered geometries.
Actually, that's a very good point, and very recent paper has come out that studies this, like maybe about a month ago or so, but exactly addresses this. So looking at, rather than hexagonal order, looks at like regular ordered geometry, and the effect is different. So it very much depends on where you have the stagnation point, and that changes how elasticity plays a role.
I see. It's quite funny. I mean, in terms of thermodynamics, one could expect that in the case of periodic distribution, you will have more flow, right? I mean, it's
so you get
counterintuitive.
Yeah, exactly. You may think that way, but again, this happens for the order geometry we studied. But if you change the order geometry rather than hexagonal array to a more checkerboard array, in that case, you would see opposite.
I see. And my other comment regarding about the biofilm that you show, in order to, let's say, to say that as a motivation, I would say that the bacteria move around this polysaccharide media, right, given by the EPS in the biofilm. But as far as I know, when bacteria are in the biofilms, the motility of this bacteria typically is very low, not because of the media itself, but actually it's a more biological thing. I don't know. I heard, I mean, I read something about that. Actually, the bacteria that born in the biofilm born without flagella. So at which point is the modeling, the movement in the biofilm is quite important, let's say.
That's a very good point. Yeah, most bacteria that actually are going to the biofilm mode, they become non-motile. But there was a paper maybe 3-4 years ago published that was showing some of the bacteria within the biofilm still have their motility, and kind of they help transfer of nutrient across the biofilm. So you would say in that application would be more applicable to those, but you're right in terms of most of the bacteria becomes non-motile.
Thank you. That is a really interesting talk. I don't know if there are some other questions. If not, we really thank you for your talk, Aresu.
Thank you. Thank you very much for the invitation and organizing the conference.
Thank you. Now we can move to the other talk that will be given by Juan Rubén Gómez Solano from UNAM University, National, I don't know how to say this in English, Universidad Nacional Autónoma de México. I don't know if you have a good translation for that, Rubén. But yeah, let's start with Rubén.
All right, let me thank you, Francisco, and let me share my screen. Please let me know if you can see it correctly.
We are seeing your PowerPoint. Yes, now is the presentation on. Okay,
great. So first of all, I would like to thank the organizers for inviting me to give a talk in this very interesting conference. In the next 40 minutes, I'm going to present some experimental results about thermally activated transitions of a Brownian particle in a viscoelastic environment. These are some experiments I performed recently at the Institute of Physics of the National Autonomous University of Mexico. So the motivation was to study thermally activated transitions because they are a very ubiquitous process at mesoscales in nature. And they happen in systems with multi-well energy landscapes subject to thermal fluctuations. For instance, this happens in macromolecular folding. In the case of proteins or DNA hairpins, they can exhibit different conformations. For example, in this special case of proteins, they can exist in an unfolded state, and because of thermal activation, they undergo transitions to a folded state or a native state, and this happens precisely because of these thermal fluctuations. So if we look at the energy landscape, the free energy landscape of the protein, then as a function of a reactive coordinate, a reaction coordinate, then the conformational changes make it go from this unfolded state to the folded state. And this happens also for DNA hairpins. This is also important for drug binding. In drug binding, basically we have a ligand and we have a macromolecular target. So the ligand must bind with the target. And for this, it has to explore a complex free energy landscape with many metastable states. And in the end, it reaches this bound state. So here it is very important to know the transition at which this process happens, because this determines the efficiency of the drug. And another system where thermally activated transitions happen is in colloidal absorption at fluid-fluid interfaces. For example, when we have a colloidal particle straddling at a fluid-fluid interface, for example, water and air, because of the presence of thermal fluctuations in the system, deformations at the contact line happen, and these deformations lead to an increase in the local drag of the particle. So this is very important to determine the properties of these systems. And all these processes, all these thermal activations have been investigated and understood in the context of Kramer's escape rate theory, which was proposed almost 80 years ago. And this theory uses a model, a very simple model, which consists of a Brownian particle coupled to a viscous environment and initially sitting in a potential well. So the particle is initially in this metastable state, which is separated by this energy barrier. But because the particle is Brownian, it is subject to the thermal fluctuations of the surrounding medium. So if thermal fluctuation is strong enough, the particle can escape over the barrier. And Kramers provided an analytical formula to describe the escape rate at which the particle crosses this energy barrier. And the formula in the case of an overdamped particle, where if viscous forces are much larger than inertial forces, has this form. It depends on this Arrhenius-like factor, which depends on the height of the energy barrier, on the thermal energy of the system, and there is a prefactor which involves several terms related to the shape of the potential, and also to the coupling of the particle and the surrounding medium. For example, in this expression, we have also the local curvature of at the top of the barrier. We also have the curvature in the potential well, and we have the friction coefficient, which is very important because the particle is not isolated, but is coupled to this viscous environment. So based on, well, as I mentioned, this model describes many of the situations I presented before. And very recently, with the advancement of optical micromanipulation techniques, Kramers' escape rate theory was precisely verified. For example, in the late 90s, an experiment was carried out by using two optical tweezers, which consist of two highly focused laser beams, and they are separated by a small distance. So in the experiment, a colloidal particle, a particle with a radius of a few micrometers, is trapped by one of these optical traps. But because the particle is surrounded by water, then it experiences thermal fluctuations, so it can jump to the next optical tweezer. And this is separated by an energy barrier, just as described by Kramers' escape rate theory. And by measuring the rate at which the particle crosses this barrier, it is possible to make a comparison between Kramers' escape rate theory and these experiments. So this was done in this paper. This was published in this paper. And it was verified that Kramers' escape rate theory actually describes very nicely this kind of processes. This was done in the overdamped case, where the particle experiences a very high friction. And more recently, by means of the same kind of optical techniques, it was also verified that Kramers' escape rate theory is valid in the underdamped limit, where inertia is important. So in this case, the difference is that the particle now is, in this case, it was a micron-sized particle. This was a nanocolloid, which was trapped by this bistable optical potential in vacuum. So the dynamics is underdamped because inertia is not negligible, and different regimes of Kramers' escape rate theory was verified. So in particular, here I'm showing a special phenomenon which was predicted, and is called the Kramers turnover. So so far, all these experiments were performed either in water or in air, which are purely viscous fluids or Newtonian. But as we know, many fluids in nature are viscoelastic. And as presented by the previous speaker, this has very tremendous consequences for the motion, for example, of microswimmers, or in general of colloidal systems. And this happens mainly because of the time-dependent flow properties of viscoelastic fluids. For example, here I'm showing again this very nice experiment where it was shown that sperm cells swim in a collective manner in a viscoelastic fluid, and this phenomenon is absent in the case of swimming in viscous fluids. Also, this happens in Janus synthetic colloids, which are self-propelled particles. When they move in a viscoelastic environment, they exhibit very interesting circular trajectories, which are absent in the case of Newtonian fluids. And all these phenomena happen basically because all these viscoelastic fluids have a complex microstructure which is elastic and is embedded in a viscous solvent. So they can exhibit both liquid and solid-like behavior depending on the deformation that is imposed on them. And the most basic feature is that, for example, here I'm showing what happens in a viscous fluid under a step-like shear deformation. In this case, the shear stress immediately relaxes to 0. But in the case of a viscoelastic fluid, the relaxation is much slower in general. So the stress under this step-like deformation decays, and in this transient regime, it behaves like a solid, but in the long time limit, it goes to 0, so it flows like a viscous fluid. And in view of all these properties, our goal was to perform an experiment of thermal activations of a Brownian particle, but in a viscoelastic environment, in order to investigate whether or not Kramer's escape rate theory is still applicable or not. And in case if we find difference, to try to explain what's the origin and related to the rheological properties of the viscoelastic fluid under investigation. So we published these results in this paper. So basically, in our paper, we reported the experiment of a colloidal particle in a double well optical potential, like the potentials I presented previously. The particle is moving in a viscoelastic fluid environment with a large relaxation time. And we try to perform a neat comparison between our experimental results and a theoretical model which incorporates the viscoelastic response of the fluid. So first of all, the fluid we used in the experiments consists of a micellar solution composed of the surfactant cetylpyridinium chloride and the salt sodium salicylate. So the surfactant, the molecule has this shape. It has a hydrophobic tail and a hydrophilic head. So when mixed in the ionized water, it forms these micellar structures. The tails try to avoid water. So that's why they form these, at low concentrations, they form these spherical micelles. And by increasing the concentration, in particular, we use 5 millimolar in concentration, they form these elongated worm-like structures, which are the so-called worm-like micelles. And the typical length scales of these systems are the following. The micelles have, the worm-like micelles have a radius of 2 nanometers, a contour length of from 0.1 to 1 micrometer, so they are not rigid structures. They form and deform in the solvent, and they have a persistence length of 10 nanometers and a mesh size of 100 nanometers, roughly. But the importance of using this specific system is that it exhibits a very simple viscoelastic behavior. So in the linear response regime, the relaxation modulus of this system, which can be measured, for example, in a rheological experiment just by determining the ratio between the stress relaxation and the imposed deformation. In this case, it has this form. It has an instantaneous response, which is related to the high frequency viscosity of the system, which is related to the solvent. The solvent is basically water. And also we have a slower response, which decays exponentially in time. So in this second term, there is a relaxation time related to the typical relaxation times of the breaking and recombination of these micelles. And we have a prefactor, which is related to the elasticity of the micelles. So then the next step was to measure the free Brownian motion of a particle suspended in this viscoelastic fluid. So for this, we used a micron-sized particle. We recorded by means of videos the motion, and then we determined the mean square displacement. In 2 dimensions, the experimental results look like this. This is the typical Brownian trajectory, and we observed that there are 2 important behaviors. At very short time scales, the mean square displacement is diffusive with a diffusion constant, which is related to the high frequency friction coefficient, which is in turn related to the high frequency viscosity of the system. So at short times, it's more or less linear. And also at very long times, the mean square displacement is diffusive, but the diffusion constant is different. It's actually smaller than this one. And in this case, it's related to the zero frequency viscosity or the zero shear viscosity of the system. And this leads to a zero frequency friction coefficient. And at these intermediate time scales, it has a non-diffusive form, which is related to the effect of the micelles, to the elastic effect of the micelles. And from these mean square displacements, we used a generalized Stokes-Einstein relation because the system here is kept at constant temperature. So by taking the Laplace transform of the mean square displacement, we could compute the frequency-dependent viscosity of the system. So the results are shown here with these turquoise diamonds. And as you can see, if we plot this frequency as a function, if this viscosity as a function of frequency, it exhibits a pronounced frequency dependence. We compared this with the results for the Brownian motion of a particle in water. So the results are shown here, and we observed that there is a very important difference. And if we use our model for the linear relaxation modulus of the micelles, then we can determine the shape of the frequency-dependent viscosity, and we can fit our experimental results to this function, from which we can compute the experimental values of the zero frequency viscosity, the infinity frequency viscosity, and the relaxation time of the fluid. So in this special case, basically our zero frequency viscosity is more or less 40 times higher than the frequency of water. Also, the solvent viscosity is 4 times higher than the viscosity of water, and the relaxation time is around 1 second. So the next step is to perform the experiment of thermal activations of a Brownian particle in this specific viscoelastic fluid. For this, we used an experimental setup based on optical tweezers. So for this, we used 2 orthogonally polarized optical beams of a green laser. They were focused by means of a high numerical aperture objective into a sample cell, which contains a suspension of the viscoelastic fluid I described before, and spherical silica particles with a radius of, a diameter of around 1 micrometer. This is kept at constant temperature, and we keep the particles at least 10 micrometers away from other particles or from the walls of the sample cell. And this is to avoid hydrodynamic interactions, which could obscure our experimental interpretation of this phenomenon. So we trap one of these colloidal particles by this optical potential. So this is a diagram of the potential. It consists of 2 optical traps separated by a small distance, less than 1 micrometer. And the particle experiences this effective potential. The potential has 2 local minima, which are denoted here as A and B, and they are separated by this barrier, which corresponds to this region where the laser intensity is lower than in this region. So then the particle undergoes the activated transitions I mentioned before. So the particle can stay here, but because of thermal fluctuations exerted by the surrounding fluid, it can jump to the next potential. And this process happens stochastically. This is what it looks like in practice. So I will reproduce this video where this is a colloidal particle, the particle I described before. This is surrounded by the viscoelastic micellar fluid. And as you can see, the position fluctuates a lot because of the thermal fluctuations. But in addition, the particle jumps from 2 well-defined positions. You can see that from time to time, it jumps from here to here. And this happens in a stochastic manner. So what we did was to record, let me close this video. We recorded videos to track the motion of the particle and then to study the thermal activation during this process. So basically we recorded the videos, we determined the particle position in 2 dimensions, because with these videos we only track the 2-dimensional particle position. And because the system in the long time limit is in equilibrium, so we can use the Boltzmann distribution in order to determine the specific shape of the potential. So by inverting the Boltzmann distribution, we obtain this kind of profile for the potential experienced by the particle. As you can see, the potential has these 2 minima. So these dark regions corresponds to very deep regions of the potential. And here there is a potential barrier, which separates both minima. I will denote these potential wells as A and B, and the potential barrier as S. So the next step was to study more carefully the dynamics of the particle in this potential. So for the sake of simplicity, we focused on one-dimensional problem. So we only studied the motion along the line which connects the 3 critical points A, S, and B. So the potential along this coordinate looks like this. Once we have this potential profile, we can fit it to a specific function. In this case, we found that the shape of the potential is described by a double Gaussian profile. And from this function, we can determine the parameters needed to make a comparison with Kramer's escape rate theory. Specifically, we need the local curvatures of the potential here at this position A and B, and we also need the curvature at the top of the barrier. And in addition, we need also the height of the barrier. And here it's important to note that in practice, it's impossible to create perfectly symmetric potentials. So there's always a very small difference between the depth of A and B. So actually, this is very useful in the analysis because we can investigate separately the transition from A to B and the transitions from B to A over the barrier S. And this is a typical stochastic trajectory during the escape process. So as you can see, the particle stays for a certain time in A, then it jumps to the wall B over the barrier. And then what we can do is to determine the time the particle spends in a certain potential well until it reaches the next well. So we can compute this time and to collect all the different values of tau for certain transitions, in this case from B to A. And similarly, we can do this, we can do the same for the transitions from A to B. So we determine the time the particle spends in the well A until it reaches a well B. And from this set of values of tau, we determine the distribution of crossing times. For example, this corresponds to the distribution of the crossing times from A to B, and this from B to A. And a very important observation of these results is that they can be described by a simple exponential. So they can be fitted to this exponential function, which only involves a single parameter. And this parameter is the mean barrier crossing time, which is related to the quantity described by Kramer's escape rate theory. So the fact that we observe an exponential behavior of this distribution simply suggests that these activated transitions are independent from each other. So this is basically a Poisson process. And once we determine the mean barrier crossing time from these numerical fits, we can directly compare with the prediction of Kramer's theory. So the results are presented in this plot. So the vertical axis corresponds to our experimental results. The horizontal axis corresponds to the prediction given by Kramer's escape rate theory. Here I'm showing you again the expression provided by this theory for a fluid with a Newtonian fluid with a constant viscosity equal to the viscosity, the zero shear viscosity of the viscoelastic fluid. So we observe that Kramer's escape rate theory overestimates the mean crossing times we observed in our experiment. So in order to check that this was an actual effect due to the viscoelastic fluid, we repeated the experiment, but in water. Instead of using the viscoelastic micellar solution, we use just water. And because the viscosity of water is much smaller than the viscosity of the micellar fluid, the typical crossing times are much shorter. So that's why they are just in this part of the plot. But in order to better appreciate the comparison with Kramer's escape rate theory, here in this local plot, we can see that there is a very good agreement between experiments and theory. So with this, we verified that our experiments in the viscoelastic micellar fluid were correct. So there is a systematic reduction of the mean crossing times in a viscoelastic fluid if we use directly Kramer's escape rate theory. So in order to understand the reason of this reduction, we used a model based on the generalized Langevin equation for the motion of this coordinate of the colloidal particle. So in this model, we basically consider the optical forces exerted by the potential, which are taken into account by this term here. And we consider the effect of the friction exerted by the fluid on the particle. But because the particle now is in a viscoelastic environment, so instead of using friction coefficient times a velocity, we use this memory kernel, which is related to the relaxation modulus of the fluid by means of this relation. And by using the specific form of the relaxation modulus, which we determine independently from the free Brownian motion of the particle, so we can determine the frequency-dependent particle mobility just by taking the Fourier transform of this memory kernel. And we observe that this mobility has two limiting values. The first one happens at small frequencies. In this case, the particle mobility is simply given by the inverse of the zero frequency friction coefficient. And in the case of high frequencies, the mobility is related to the inverse of the high frequency friction coefficient. And finally, in the model, we also consider the effect of thermal fluctuations because they are necessary in order to understand the thermal activations. So for this, we use this stochastic Gaussian force with zero mean, and which is related to the memory kernel by means of this relation. The autocorrelation function of the stochastic Gaussian force is related to the memory kernel by means of the second fluctuation dissipation theorem. So now, once we have this model, we can follow an approach based on Kramer's work, but by taking into account an arbitrarily long memory in the system. So the base, well, actually this was proposed long time ago in the 80s. And the basic idea of this approach is to linearize this equation of motion around this energy barrier. Because with this, we will consider the escape process of the particle during a thermal activation. And from this, we can obtain an effective Smoluchowski equation for the probability density function of the particle around the barrier. And two important quantities can be derived from this Smoluchowski equation. First of all, we can determine the non-equilibrium steady state probability current for the particle to go from here to here. And second, the occupation number, because this is related to the probability of the particle to be here initially in thermal equilibrium in this potential well. By taking the ratio of these two numbers, we obtain directly the mean time for the particle to cross the barrier by thermal activation. So the important result of this analysis is that we recover this expression, which contains this Arrhenius-like factor, which also appears in Kramer's escape rate theory, but with a prefactor which now depends on the Laplace transform of the memory kernel of the system, because now the particle is coupled to a viscoelastic environment. And it also involves the local curvatures of the potential well and the curvature at the top of the barrier. So if we do the calculations for the specific shape of this memory kernel, we obtain the following expression. We can express the mean crossing time of the particle in the viscoelastic fluid in terms of the mean crossing time of the particle in a viscous Newtonian fluid with the same zero shear viscosity as the viscoelastic fluid times a prefactor, which is a correction to Kramer's escape rate theory. And for the specific form of the memory kernel, it has this, this expression. I will call this corrective factor beta, and it depends on two interesting parameters. They are non-dimensionless. The first one is alpha, the ratio of mobilities, the ratio of the zero frequency mobility of the particle compared to the frequency at, to the mobility at high frequencies. And it also depends on the ratio of the viscoelastic fluid, of the time scale of the viscoelastic fluid, and the time scale the particle needs to undergo dissipation during the escape over the barrier. So this is a very nice expression. It's very simple, but it explains what we observe in the experiment. So first of all, we checked that our expression reduces to Kramer's escape rate theory under certain specific limits. For example, if we take alpha tends to 1, which corresponds to a situation where there is no frequency dependence of the mobility. In this case, beta tends to 1. So we simply recover Kramer's escape rate theory, because this is 1. So this is expression provided by this theory for a Newtonian viscous fluid. And second, we can also consider the limit when tau 0, the relaxation time of the fluid, is very short compared to the slowest dissipation time scale of the particle in the neighborhood of the barrier. So in this case, this also corresponds to a Newtonian situation in which the relaxation of the fluid takes place on a very short time scale. And in this case, also beta tends to 1. So the expression, with this expression, we recover Kramer's escape rate theory, but in general, we account for the effect of the coupling of the particle with the viscoelastic environment. So now the final step is to compare our new formula with this corrective factor with our experimental results. So this is what we've had before for Kramer's escape rate theory and the experimental mean crossing times. And this is by taking into account this correction by means of the prefactor beta. So in this case, we observe that there is a very good agreement between our experiments and these new corrected mean crossing times. So now we can, well, first of all, in this plot, I'm showing you the experimental values of beta for our experiments in water and in the viscoelastic fluid. This solid line corresponds to the analytical expression provided by this formula. And what we can observe from here, well, in water, as expected, beta is very close to 1. And this can be understood by the fact that in water, we have alpha equals 1. The mobility is independent from the frequency. And the relaxation time of water is also very short. For our practical purposes is 0. So we are in this part of this plot. But in the case of the transitions in the viscoelastic fluid, we find that beta is rather small. It's around 0.2. So there is a very large reduction of the transition times. And this happens because the values of this ratio, the relaxation time of the fluid compared to the viscous relaxation time of the fluid on the top of the barrier are similar. They are around 1.2, 1.7. So with this, we can understand the reason why this reduction happens. And this is basically because when the particle undergoes this stochastic transition over the barrier, the fluid does not have enough time to fully relax once the particle reaches the neighboring potential well. So this is an unequilibrium process during the escape. And well, this provides evidence that this is related somehow to the transient nature of the mobility of the particles. So in order to check this, what we did is to compute the effective particle mobility in different regions of the potential landscape. For example, we can compute the particle mobility in the potential wells, A and B, and we can also compute the mobility over the barrier. So for this, we just simply determine the ratio between the mean particle velocity divided by the mean optical force exerted by the optical potential. And our experimental results are the following. So here in this plot, I'm showing you the effective mobility at different locations on the energy landscape. So this corresponds to regions around the well A. These are around well B. And what we observe is that here the mobility is very small. And if we compare the typical experimental values for this effective mobility, they are very close to the values that correspond to the zero frequency mobility. So basically here, the particle is experiencing the mobility in the long time limit. But when we compute the mobility during the escape process over the barrier, which corresponds to this location here, we observe that the mobility increases a lot by 1 order of magnitude, and it's very close to the value at high frequencies, to the mobility at high frequencies. So basically here, the particle is only feeling the short time response of the fluid. So yeah, and this is related to this observation I mentioned before. The particle crosses here without experiencing the total friction given by the micellar structure. So just to check this, we also computed the effective mobility during the transitions in water. What we observe is that in this case, the effective mobility is rather constant and is simply given by the inverse of the friction coefficient of the particle. Finally, we perform another analysis in order to check these results. So they are based on the transition path times. So the basic idea is to compute the time the particle needs to go from a certain position over the barrier and to reach the other side of this energetic barrier. In the case of water, this was checked recently in this paper. And in this special case, when the particle is surrounded by water, the transition path time distribution has this special form. Now it's not exponential anymore because this is a different kind of process compared to the previous escape process I presented before. And it has a more complicated shape, this distribution. It's basically, it goes to 0 at short transition path times, it has a maximum, and then it decreases to 0. And well, in this paper, the authors checked that actually the transition path time distribution has this specific shape. So we performed the same analysis in our experiment in a viscoelastic fluid. So for this is a typical trajectory during the stochastic transitions in a viscoelastic fluid. And I'm showing you in different colors the transition paths, which are different from the trajectories I showed before. Before, what I computed was the total time the particle spends in a certain well, and then until it jumps to the next one. Here, we are only interested in the time the particle needs to go from a certain well to the other, which are shown here. So because now we have a memory kernel, which can be very well characterized because we determined this from these microbiological experiments, we can obtain an analytical expression for the transition path time distribution in a viscoelastic environment. In this case, it's much more complicated than this expression, but still it can be computed. So now the difference is that it involves here this function chi, which is related to the Laplace transform of the memory kernel. This is a function we know from the experiments, so we can determine the shape of this theoretical prediction. And this is what I'm showing here with this blue curve. The histogram corresponds to our experimental transition path times. So we observe that there is a very good agreement. So this only confirms that our theoretical model is valid, is applicable to this viscoelastic environment. But what we can do now is to compare to the transition path time distribution for a particle in a Newtonian fluid with a zero frequency viscosity equal to the zero frequency viscosity of the micellar fluid. So we observe, this is shown here with this red curve, and we observe that there is a very, very large difference between these distributions. But when we compare with the transition path time in a Newtonian fluid with a viscosity equal to the solvent viscosity, to the high frequency viscosity, we observe that our experimental results and our theoretical formula is very close to this. This is shown here with this green curve. So with this, we confirm that actually during the crossing process, the particle is only experiencing the high frequency friction, mainly due to the solvent. So with this, I would like to summarize my presentation. We performed the first experimental realization of thermally activated transitions of a colloidal bead in an optical double well potential in a viscoelastic fluid. The main observation is that there is an amplification of the transition rates induced by viscoelastic memory friction. And so the typical mean crossing times are much shorter than those predicted by Kramer's escape theory for a similar Newtonian fluid. So we perform a precise characterization of the potential, of the optical potential, and also of the viscoelastic properties of the fluid. And from this, we could make a very neat comparison between experiments and theory. And we also provided direct evidence of the origin of the phenomenon. And this is mainly due to the transient particle mobility, which increases around the energy barrier. So as an outlook of this work, we are currently working on the same process, but in a periodic potential landscape. In this case, we don't have just two local minima, but we have several minima over a periodic extended landscape. We would also like to study this effect in active matter. For example, with Janus colloids. Here the difference is that instead of having thermal activations, these transitions will be triggered by the activity of the particles. Also, we are working on time-dependent potentials. This is also very important in the context of stochastic resonance. And we also plan to do this in order to understand complex density of materials. And finally, I would like to acknowledge my collaborators, Brandon R. Ferrer, who is doing his master currently under my supervision, and he performed most of the experiments presented in this talk, and also Alejandro Vázquez Arzola, who is a researcher also at the Institute of Physics at UNAM, and he assisted me with many of the experimental details of the setup. So thank you for your attention.
Thank you, Ruben. Really nice talk and all this summary of your experiments now in your lab in Instituto de Física. If there are any questions from the audience, I think Eric is raising his hand. Please, Eric, go ahead.
Thank you, Paco. Thank you, Ruben. Such a nice work. I was wondering, I see that the distribution of jumps time between the A, B, and B and A are slightly different. I mean, you compute from them the time required, or the mean time required for the particle to jump the barrier. Yeah. It seems to be slightly different. I mean, for example, for the first bin, in one case is slightly above 0.01 and 0.4, and the other case is slightly below. Is that statistics, or maybe related to what you mentioned before, that the optical traps are slightly different in some cases?
Yeah, this is fully correct. So there is a difference in the distributions of crossing times, and it's because of the shape of the potential. So as I mentioned, there is a slight difference between the depth of A and B because of, creating a perfectly symmetric potential is impossible in practice. So even a small difference can lead to a rather large difference in distribution times. And now we understand this because of this. This is the case of Kramer's escape theory, but even in our experimental expression we obtain, there is an Arrhenius-like dependence on the height of the potential barrier. So this leads to, they are still exponential in both cases, but the parameters are different for transitions from A to B compared to those from B to A. So yeah, this is basically due to the asymmetry of the potential.
Okay, yeah, and I can see that a slight asymmetry can have a very important impact on the distribution, right?
Yeah.
Okay, I'm sorry, Paco, if I can continue with my next question. Kramer's theory determines mainly the time that a particle requires to reach a certain point, as you mentioned, for example, in your previous slide, between the bottom and the top of the potential, but not the crossing time itself, because in some cases the particle can actually return. I mean,
Yeah.
reach the boundary, the saddle point, and then go back. It's difficult to experimentally, for example, capture those events where you actually reach the saddle point, and that actually counts for Kramer's time, but not for barrier jump time.
Yeah, under the experimental conditions here, they are very rare. We barely observe them. Maybe this one would be, here you see that the particle is sitting in A, and then it reaches S, but it returns. It comes back to A. But, well, actually, this is just a portion of the trajectory. So typical trajectories for each histogram, they are roughly 40 minutes. So they are much longer than this. They don't really change. They are very rare. That's what I'm going to say. Most of the events consist of these well-defined jumps. The particle stays here, and that it spontaneously jumps to the next potential well, and there is no recrossing.
Yeah, I can see it.
Yeah, it could be interesting to specifically focus on these events, for example, and maybe to analyze them separately. But they are not very common.
Okay. Yeah, I see. Actually, your trajectories are very confined in the tweezers. Well, in the minima, right? And then the jump is quite obvious. Okay.
Yeah, and this is also a very interesting problem because here the requirement for this theory to be valid is that the height of the barrier is large enough, typically several times kBT, the thermal energy. Because if the potential is very shallow, if here we have a very small barrier, typically less than 1 kBT, then these events are much more common. But in that case, we cannot follow this approach because this approach requires that we start from a perfectly initial equilibrium state. The particle stays here, it's equilibrated here, and that's why we can compute this occupation number in equilibrium. But this is not true if the potential is very small, if the potential barrier is very small, because the particle does not have enough time to equilibrate here, so it jumps before it reaches equilibrium. And that's another problem. It's actually very interesting because I imagine that there are many situations where the potentials the barriers are not large, but they need a different analysis.
Yeah, no, actually, I've seen a lot of papers, for example, in the last 10 years or so, where this problem of, let's say, first mean-field passage time is now being analyzed, for example, theoretically by using the Smoluchowski equation or so, and then they compute not only the mean value but also the distribution of times. are very awesome works actually. Some of them it seems that can be experimented and realized by optical tweezers.
Yeah, definitely. That's one of the aspects we actually wanted to go into this direction, to study not only the escape process, but also these transition path times, which are related to these inverse passage times somehow, we forget about the dynamics here, and we only focus on the dynamics from a certain point on the barrier to another point. And this is true. Now the distributions are not exponential. They are much more complicated. And yeah, so it would be very interesting to analyze this even in the case of small potential barriers, because things are totally different. So the assumptions required for the derivation of these expressions are not valid anymore.
That's it. Thank you so much, Ruben. It was a very nice talk.
Thank you.
Thank you, Rick. Is there anybody to ask a question? Nope. If may I, Ruben, to ask a couple of questions. I have a curious. I'm wondering about the experiment. I'm not expert in experiment, but I'm wondering about this potential depth that you mentioned, that you have this asymmetry in the potential wells.
Yeah.
So it recalls me that maybe what you have there would be a ratchet effect, maybe due to the asymmetry, and maybe you will have more events that the particles move to one well to the other instead of the other way because of this asymmetry.
Yeah, so
it looks like a flow, no? I mean, like the ratchet movement.
Here, the system is in thermal equilibrium. We are not applying any external force. So in the long time limit, of course, this will translate into different mean crossing times because one of the potential wells is deeper. But in the end, the system is still in equilibrium. So there will be no net motion of the particle. So if you compute the mean particle position, will be somewhere here. So there is no.
I see. And is it difficult to you to tune the depth of the trap?
Yeah, it's somehow difficult because these experiments are very sensitive to the shape of the potential. So actually, here the potentials, the optical traps are separated by around 700 nanometers, but by changing slightly, the potential disappears, the double well potential disappears. So there are many, this is related to the specific form of the optical forces acting on the particle.
I see. I thought it was related to the power of the laser. I mean the depth of the book.
Yeah, with the laser power, but we can change is the depth, for example.
Yes.
Because the depth is related to the optical trapping force, the local optical trapping force. So higher intensities, higher laser powers give rise to deeper potentials, but the shape changes a lot. So this is actually, that's the reason why these experiments are very, very difficult to realize.
Yeah, because I found quite interesting the question that you mentioned. I mean, when you answered the question of Erika about when this regime is valid, right? And you mentioned it's something like several KBTs so I was wondering if it's whether possible to modify the depth, no, like systematically in order to see this transition in the regime of where it's valid the model.
This could be possible, but actually that's one of the problems we are interested in here at the Institute of Physics. And it's the problem of extended potential landscapes. Instead of having a single or just two potential wells, to have an extended periodic potential, like a substrate. And then the problems you pointed out could be investigated. For example, the motion, the directed motion across a periodic landscape. For this could be created with a tilted potential, for instance, or to apply an external force which bias the the motion. And actually the motivation is this, to study the transport of particles across an extended landscape with potential barriers. But in addition, we have the effect of the fluid. The fluid is not, the coupling between the particle and the fluid is not purely viscous, but it exhibits this heterogeneity. Depending on the position across the potential, the particle mobility is different. So actually, the mobility, this is somehow related to what I mentioned here. When the particle experiences this transition, the mobility increases, which is different from the motion in a purely viscous fluid. So this must have very tremendous consequences in extended periodic landscapes. Yeah, but this is still an open problem. We are working on it.
Yeah. And regarding this mobility, that's the other question that I have about this viscosity that you measured there in terms of the mobility, this view. It recalls the previous talk of Aresu. I don't know at which point this difference in the interface, it has to be with maybe with the alignment of the polymer and the polymer melt that you have there. I mean, the viscoelastic fluid that you have there. Probably this difference in the mobility in the interface would be since you have thermal activate the fluid with the laser, you modify the structure, I mean the microscopical structure of the polymers in the fluid. It has sense that I don't know.
Yeah, this could be the interpretation that basically the particle during the transition is very fast. That's why I showed these plots here. So the typical time the particle needs to cross just to perform this crossing is around less than 1 second. But here, this is the important point of measuring the relaxation time of the fluid. So the typical relaxation time is 1 second, 2. So the particle crosses, but the fluid has not yet relaxed. So this is somehow to this observation pointed out by you. So the fluid is deformed, the micelles just undergo some deformation, but in the meantime, the particle is already here. So that's why, because this process is very fast, then the particle is not totally subject to the low frequency response of the fluid. So it only experiences the viscosity or the friction done by the solvent. Here, the fluid, maybe it's related to this. The fluid consists of micelles, but these micelles are in a solvent. So the particle, the friction is only related to this solvent viscosity, or mainly, not totally, but mainly.
Quite interesting. Thank you, Ruben. If there is no more further questions from the audience, yes, Eric, please.
Sorry, I lowered my hand and then I raise it again. That's
Do you have any other question now? You just mistake in the button.
No, I have it. Okay. When you were discussing actually with Paco, you showed the image of the optical traps, and then remind me something. In that case, I mean the optical stiffness in the direction perpendicular to the ray. I mean in that direction should be
this one.
Yeah, exactly. In that direction, in the direction of that could be the vector wave. In the direction of the optical stiffness is slower actually than in the other cases. You see that maybe the particle moves slightly to in that direction just to have maybe a better transition, or there is no the focus on the particle when the transition takes place?
Not really. Of course, there is motion also along this direction, but well, I can show you the typical videos. The particle looks like this.
Yes.
So it
the
also
Z
moves.
Direction.
Yeah, the Z direction in this case is perpendicular to this plane.
Okay.
But here, which is important maybe, is that the particle is made of silica. So also it's trying to sediment, but it's kept by the optical potential. It does not go to the bottom of the sample cell. So this prevents that the particle moves much to the upper direction. I don't know if this.
Okay, yeah. No, I mean, you can clearly see that there is no defocus during the transition. But I was wondering that in some case, under particular circumstances, maybe a slightly a higher energy barrier would be easier to actually jump. I mean, perform a displacement in the set of reaction, in this, up, way, down, and jump to the other track. But that could be a completely different scenario.
Yeah, definitely. And this is especially important for very small potential barriers. This is one of the technical problems. We are discovering that if we want to perform these experiments for barriers less than 1 kBt, then the optical trapping force will be very small. So the motion along this set direction will be important. The detection, it will be maybe a problem, but this could also lead to a very interesting problem, because now the potential is not only 1D, but we have to take into account the full 3-dimensional dynamics.
Yes. More complexity, more fun. Thank you.
Thank you.
Okay. Is there any further questions from the audience? If not, we can end the session, for the morning session, and we returned at 11:20 with Francisca and the poster session. Okay, so this session will be still open if you want to talk with Juan or with other attendees, panelists, and yes, see you in a couple of minutes. Bye. Is there any announcement, Eric, Victor?
As far as I remember, we only have 3 or 4 videos for the poster session. So
yes, we can discuss the posters, no? Maybe with some attendees that have posters. Maybe we can open the mic for them. What do you think?
Or we can invite also to other attendees that are presenting posters that maybe want to just not copy. I mean, not reproduce the video, but yeah, discuss the poster. Yeah, you're right. Perfect. That would be great.
Okay. See you in 20 minutes. is better. So we'll change to English.
Switch,
Francisca, please.
please.
Okay, yes.
So maybe shall we start, Victor?
Who?
Shall we start now, or we wait a couple of more minutes?
If you want 2 or 3 minutes, whatever you want.
Let's wait a couple of minutes. Okay, let's start the second session of today. We will start with Francisca Guzman. She's from Universidad Mayor from Santiago de Chile, and she will talk about active carpets, learning to transport at the microscale. So whenever you want to start, Francisca, thank you.
Thank you, Francisco. So yes, thanks for the introduction. Today I will talk you about an area or that we, with my 2 colleagues, have been exploring during the last 4 years. So this is a funny work and a funny history behind it. So I was doing my postdoc with Hartmut Leuven in Düsseldorf, Germany. And when I was finishing my postdoc, I met Arnold there. And we, like, in one afternoon, we just write down what we want to explore. And we started right there. And we have been working with Arnold like in online way from then, so from 4 years ago. And we have met each other during the years in order to write down some papers and so on. So today I will talk about this work. Everything has been done at long distance between us. So one person has been in Germany, which is Hartmut, another person, which is Arnold, in United States, and I was working here in Chile in the same project. So the outline of my presentation is the next. So today I will talk to you about active matter, and then we will establish what is it first, and then we will talk about self-organization at all length scales, life at surface, active carpets, and we will talk about how they get nutrients, and finally we will talk about active transport. Okay. So what is active matter? So if you look at Wikipedia, you will find the next definition of active matter, which is active matter is composed of larger number of active agents, each of which consumes energy and to move or exert mechanical forces. And due to the energy consumption, these systems are inertially out of equilibrium, or thermal equilibrium. So it's very interesting because what is this definition is saying is that, okay, we can study any active agent at any length scale, and for definition, an active agent is just a self-propelled particle that can consume energy from the medium in order to move or to exert forces or etc. So here we can see this diagram where you can find the length scales where you can find active matter. You can find them in the nanometers with biomoleculars. And then you can move to micrometers to find bacteria, algae, and Janus particles. And then you can move to millimeters where you can find other microorganisms like this one in the video, which has some cilia that you can see that it's agitating, and you can see that by moving its body can move the flow around it. And then you can move to the meters, and you can study with in the same area some animals, fishes, and birds, etc. What is important from this area that we will study in this presentation is that it's a discipline that lives in the border between physics and biology. That's why it's quite fun. So what was for me or for us for this project our main inspiration, and what we wanted to study in the beginning of the project was, okay, we know that active agents or active particles moves on surfaces, okay, and we were thinking in the beginning only about bacteria. So we were thinking, okay, bacteria is moving, a lot of bacteria is moving on a surface, and we know, as you can see in this video, that while they are moving, they are forming some patches, clusters, they are self-organized, or they are moving, doing some collective phenomena. So what happens with the fluid above them? Because you know that bacteria is moving in fluid media, then what happened with the fluid when they perform this kind of self-organization? And in order to understand this, you have to first modulate your problem, and that's what I do. So when you observe a biological phenomena, then you have to write down your motion equations, or you have to model what you are observing, okay? And in order to see what is the main things that you want to capture, I will talk you about some ways to modulate or to modulate active matter. So the first thing that you can observe in this video is that bacteria is using, or it has a a specific volume or space. So bacteria is like a solid particle, and then you have to say that they are have some exclude volume interactions between them, okay? And then you have to write down an equation for this exclude volume in order to prevent this type of interactions between them. And also bacteria, when they are moving in the fluid, they can create some distortions in the fluid. These distortions are long range, and they are called hydrodynamic interactions. And due to these hydrodynamic interactions, a lot of collective phenomena can emerge in the system, such as synchronization. So in this video, the second one, you can observe this vortex-shaped bacteria, and they are all synchronized, okay? The vortex. And the synchronization is only due to the long-range hydrodynamic interactions between the agents or between bacteria. And then you have to write down an equation for the hydrodynamic interactions that you want to take account, okay? And finally, in another scale, and here are actually two different scales. In the scale of the millimeters, we have the ants, and for ants, you know that, okay, ants are not moving in a fluid media. Nevertheless, they are still in the field of active matter. So for active matter, it's not necessary that you are moving through a fluid. And you can see that they are self-organized, and they are like moving again in this vortex shape motion. And in another different scale, so in the meters scales, we have this mousse. They are like confined in a place, and therefore they start to move in this vortex way that you are observing here. And if you wait a bit more, you can see that this is a huge vortex, but in the left hand side, there is a bigger one. Okay, and this is only due to the confinement, you see? And then what interactions can you put between these different organisms? Is the quorum sensing. And then you can write down a different equation to modulate the interactions between them. Okay, so this is the first part. So in this work, we are focused on the hydrodynamic interaction. So I will use for now and so on these equations in the problem. So let's start with a flagellate organism or a bacteria, and we can write down equations for the bacteria, but we can say some characteristics about them. We can say that, okay, the bacteria is a rod-shaped organism, that it's the length, characteristic length is more or less 6 micrometers, and it moves with a self-propulsion velocity of 20 microns per second, more or less. When we study bacteria close to non-slip surfaces, bacteria behaves really, it gets attracted to the surface, and when it's attracted to the surface, it starts to move in circles. So bacteria perform circle swimming in non-slip surfaces. they are also accumulate on the surface. So you can see here in this experiment, they put a drop filled with bacteria between two plates. And after some time, they observed that the all bacteria was accumulate on the surface of the two plates, as you can see here. So there are two peaks, and there is almost no bacteria between the two plates or in the bulb, let's say. And third, bacteria also, or algae or microorganisms, they perform chemotaxis. So this is the attraction to some chemical signal. And due to this chemical signal, they are also accumulating in some hotspots, okay? And so what we want to keep in mind in this part is that bacteria interactions with non-slip surfaces inhibits exploration. So they tend to accumulate, to do or to perform circle swimming. Therefore, they start to form clusters and or patches in the surface, and yes. So here I'm also showing you two plots where you can see how is the flow field of a single bacteria. Okay, in this case, the bacteria is the E. coli. So this is a puncture-like swimming. So it means that they push fluid in the front and in the back of their body, and while in the two sides of their body they are pulling fluid, as you can see here. And this is a picture of a bacteria close to a non-slip boundary, which is just here down. Oh wait, it's up now, I think, in this picture. And this is how you can write down or how can you observe the flow with equations. And sorry, here in F, this is with the equations. And here you can see how it looks in the experiments. So treat bacteria or the E. coli as a puncture microswimmer is perfectly fine. Okay. Okay, so how is the life on surfaces at the microscales? So you know that we already know that biological activity is highly concentrated on surface, where bacteria can perform some orientational gradients. And what is an orientational gradient? So we will call this when bacteria is like in a nematic order at some places, and then you can see, like right here, you can see they are aligned in one direction, but here they are aligned in a different one. So you can, in a small space, you can see different different orientational gradients. Okay, they can also perform some spatial gradients. So there are places where the bacteria accumulation is high, while in others it's not that high, and therefore you can see that they form some clusters or patches. Okay, so actually in this problem here, the different colors shows different bacterial accumulation, and you can see that in this surface, bacteria also form some places where there is no bacteria. So there are some holes in the surface without bacteria, and then you can see that there are other places where bacteria is more accumulated than in others. So now is the time when we defined the active carpet. So for us, we will call an active carpet as a non-slip surface with let's say with active agents that are allowed to move only on the surface. Okay, so for us, we would put a surface and some micro swimmers on the top at some distance from the surface, and we will allow the micro swimmers only to move in 2D. Okay, and this is for us the active carpet. But this is the first definition that we will do about active carpet, because then we will move to another microorganism like Porticella, which is also accumulated on surfaces, as you can see here. But you can observe that the motion that the Porticella is doing is a vertical motion. So it's like, you see, they grow and then they shrink like fastly. So they are doing some kind of pulses on the fluid. And we can also then study the cilia problem. So cilia for us is just so cilia for us is it's moving like a rodlet motion. Okay, and you can see that cilia can, in this case, is super synchronized, but sometimes cilia are not that synchronized in the motion. And also, of course, we will study bacteria accumulate on a surface, like in these other movies or images here. Okay, so first, let's start with a micro swimmer, which is a pusher for us, and it's we are studying in this case bacteria like E. coli, okay, moving on a surface. So first, I want to show you that, okay, when a bacteria is moving close to a non-slip surface, they have in the flow field that they create two areas, as you can see here in red, where they are pushing flow or they are creating a repulsive flow, while here in the middle, they are creating a blue flow, which is attractive, which is an attractive flow, okay? So each microswimmer for us will be modulated with the Blake tensor, okay? So the hydrodynamic interactions that we will perform are described in these two equations, where here is the flow field created by one microswimmer, which is a dipole flow at some direction P. And since the microswimmer is moving close to a surface, the tensor that appears here is not anymore the Stokes-Let, and now it's the Blake tensor, which is more complicated. Okay, but if you plot this flow field for one microswimmer, you can get this image. Now what we want to do, now we want to define a carpet of many hydrodynamic actuators. So we will put all these microswimmers like this on a surface, and we will allow them to move only in 2D, so they can only explore the surface. Okay, and as I told you, we want to know how this carpet alters the fluid above them. Okay, so how are the fluctuations that these microswimmers create vertically upon the surface? Okay, so we will measure the flow fields from all the hydrodynamic actuators in the surface at different heights far from the surface. And here is a picture of a tracer particle, and you can see how it fluctuates only due to the hydrodynamic distortions. Okay, so what we will do, so we will measure the mean flow at different heights, for all the microswimmers on the surface and given a specific architecture for the carpet. Okay, so what is for us the architecture of the carpet? So we will see now. So as I showed you before, we observe that when microswimmers are close to the surface, they tend to create density gradients and also orientational gradients. So for us, the architecture of the carpet is related with that. So now we will put different observed distributions of microswimmers, and we will measure the mean velocity or the average velocity at different heights. And this is what we get. So first, we study the case of a patch of microswimmers. So we were thinking, okay, what happens when bacteria creates these clusters? And then we get this. So we have a circular cluster in green with bacteria moving randomly on this cluster. And in gray is the surface where there is no bacteria. So bacteria is only in the gray area. And if you measure the average flow above the carpet or above this patch, you will see that clearly bacteria is creating an attractive flow or a drift, okay, that goes toward the patch, and just in the interface it creates a repulsive flux, okay. And then we were surprised about this, but if you measure with the equations, you can get actually the drift for a patch of pusher like swimmers, okay. And then if you plot this drift, you can see very interesting things. So you can get first that if you are increasing the size of your cluster, the size R of the cluster, you can find an optimal size for pulling particles towards your patch, okay. And then if you increase enough the size of your patch, at some point this drift will disappear or it tends to go to zero, which is something that we expect, since if all particles are moving randomly in all directions in the surface, then if the surface is large enough, then the mean velocity should be zero, okay. And here, so here we plot again the mean velocity, and now at different heights far from the patch. And there is also another peak. So at distance more or less like 10 micrometers, the flux is larger than in the other at a different distance. And yes, so this is what we get when we study clusters in the surface. Okay, so the message here in this first work was, okay, if bacteria wants to get nutrients, then it should be organized, okay? It should be maybe forming clusters like in this movie. So the black points are bacteria moving in the surface, and we put some chemical attractant here, and then bacteria is moving towards the chemical attractant, and it's forming this cluster, okay? And when bacteria forms this cluster, you can see that immediately the nutrients, which are suspended above the surface, tends to drift towards the bacteria. Okay, so this is the first thing that we found, and it's really nice. And then we ask ourselves, okay, it's only for the patches, and the answer is no. so the attractions of the nutrients towards the bacteria accumulation is independent of the shape of the bacteria on the surface. So then we will put this shape where the black dots are bacteria, and you can see that again in blue is the attractive flow towards the carpet, and in red is the repulsive flow from the carpet. And you can see that again, where there are bacteria, there is this attractive flow. Okay, and this is not, and if we observe these birds, they are also trying to eat by creating this special flow. And actually it's really fun, because at the beginning we were studying circular swimmers. So we were putting the micro swimmers moving in circles on the surface, and one micro swimmer is also creating this attractive flow by moving in circles on the surface, just like the birds. Okay, and here there is another picture where we put black dots are bacteria, and so the green area is now attractive flow, and the pink area is repulsive flow. And then you can maybe understand why, like when you have a dense suspension of spermatozoa or sperm, they start to creating this vortex on the surface. Then you can see, okay, if you have like a really dense carpet of sperms, let's say, you know that oxygen and nutrients tends to be really consumed in the area where they are moving, they are allowed to move in. Therefore, maybe they create these vortex arrays in order to pull more nutrients from above the fluid, you know, and in this way they can continue living like in this dense carpet. And okay, so this effect for the drift, it doesn't only appears when you have density gradients. It also appears when you have orientational gradients. So whenever the bacteria is self-organized, moving in some direction, or creating maybe a vortex, or maybe creating topological defects, you will find this attraction towards the bacteria. So, and we did then this exploration with bacterial turbulence, where you have a lot of or different topological defects. And when these topological defects, like the ones shown here, appears, you can observe that the drift, the attractive drift appears. So for us, the message was, okay, if you want to eat in a very crowded environment, then self-organize, or do whatever you can do. You can maybe create orientational gradients, or maybe you can create density gradients. So this was the first part of our work, and this is the first part of the talk also, and that was the message. Nevertheless, we were interested in the beginning in the vertical transport due to the accumulation of bacteria on surface. And then after we found this drift, we said, okay, now let's study a infinite carpet with bacteria moving randomly around the carpet in 2D again. Only they are allowed to move on the top of the carpet. And let's see if we can get the diffusion, the vertical diffusion. Okay. And in this work, we will not only study bacteria as in the other one, and now we will add another microorganism. Okay, so we will study here a carpet with molecular motors, okay, like these ones. And the molecular motors create a flow field, or they can be described by the Stokes flows. Okay, and the flow field looks like this in the photo. And then we can also study maybe the Vorticella, which is this microorganism that like elongates and shrinks with some pulses. And the Vorticella can be modeled with a Stokelet, a vertical Stokelet. And then we can also study now an infinite carpet of dipole microswimmers or bacteria. And we will also study an infinite carpet with cilia that can be modeled as rodlets. Okay. And we will measure the diffusion coefficient or the vertical transport for these different carpets. Okay. And the way we want to do this is the same way as in the before in the first paper. And now, since the average velocity it's 0, because now it's an infinite carpet, we are now interested in the fluctuations. So we will look for the variance, the fluid the flow variance or the velocity variance of the carpet. And we will do it in the same way. So we will take the different flow fields created by a single active agent, and we will put now an homogeneous distribution of them on the carpet, and we will see what we get. And here are the results. So if you study different carpets with horizontal Stokelet or vertical Stokelet, dipoles or rodlets, here are the flow fields that we will use. All of these flow fields are related with the multiple expansion of the Stokes equations at low Reynolds number. You can see that the mean velocity is 0 in all the cases, and the variance is different, okay? And which is nice is that the variance actually decays in the vertical distance from the carpet with different behaviors, depending on the active agents that you are studying, okay? So here we plot the variance for the different microswimmers types. And you can see immediately that in the 3 first cases, that the variance in the plane or in the XY direction is the same, but it's also is smaller or weaker than the variance in the vertical direction or above the carpet. Okay, so the first message is okay. The vertical transport is different than the horizontal one when you have these 3 first type of microswimmers. Nevertheless, when you are studying rodlets or cilia, the transport is exactly the same. It's independent of the direction. So then, due to the variance, we can make we can relate the variance with the diffusion, and then, as we can expect, the diffusion is anisotropic, and we can find actually also an expression, an analytical expression for the vertical diffusion for these carpets. And here we will study the vertical diffusion for the molecular motors or for the horizontal Stokelet and for the Vorticella or the vertical Stokelet. why we didn't study the dipoles, and if you check the paper, you won't see the more results about the dipoles, and it's because dipole microswimmers, since bacteria is moving in the surface, and it's not attached as the Vorticella, for example, it has at least 2 memory times, and the memory times are really were really difficult to find analytical expressions for the diffusion coefficient or the diffusivity, vertical diffusivity with these times, with these memory times. So for us, it's still, for dipoles, it's still like an open question to study the vertical transport, since it's really sensitive to the memory times that you choose in your problem, okay? But if you study Stokes-Letts, it's more easy, and we found analytical expression for the diffusivity, and we will now use only these 2 types of carpets, like a carpet of molecular motors or a carpet with Vorticella. And if we, since the diffusivity is now anisotropic and is space dependent, the fixed law for the transport, it's not anymore the fixed law, because for the fixed law you have a constant diffusion. But then you say, okay, then you have the Fokker-Planck equation, okay? But here we started with the Fokker-Planck equation for where we have like an advective term for the vertical transport of particles, okay? And that advection is only due to the induced fluctuations in the media due to the carpet. And we have also the diffusive term in the equation. But in our case, we found another different term that I will explain now. And this is the equation that we found. And the other term that appears in our equations, it's related with these hopping effects. Okay, so now I will explain. So what we did, now we put our carpets with our active agents moving in the carpet, and we will put some tracer particles above the carpet. In the first place, we will put tracer particles that have different sedimentation velocity. So this can give us an idea of particles of different sizes. They are going down due to gravity through the fluid. And due to the active carpet is there, and it's creating all these fluctuations that are strong, as we saw before. The particles above the carpet start to move like this. They are starting to agitate. And at some point, we can expect that, okay, due to gravity, these particles will like settle in the surface, right? They will settle in the surface at some point if you wait enough. But the answer is no. Due to the activity of the carpets, they create a hopping effect where particles are like suspended at some distance from the carpet. And the different, and depending on the velocity at which the particles are going down to the surface, you can find different accumulation points or at different heights. So you can imagine, okay, these particles, tracer particles, they have some sedimentation velocity, and they are hopping at this distance from the carpet. But if you put more lighter particles or particles that are moving with a smaller velocity, they will hop at larger distance from the carpet. Okay, so as you can see in the maybe in this figure, like there is a strong accumulation at some height far from the carpet. And then if you do different sedimentation velocity, as you can see here, you can find the law behind this hop effect. Okay, so the message in this work was the next. So it was, if you want to transport particles in the micro scale, then first you don't have to put like if you want to transport particles in the micro scale, don't be organized. This is the first message, because if you organize, then you will have this drift towards your microorganism. So if you want to transport things, then only move randomly on the surface. Move randomly on the surface and don't self-organize. So I found this movie on internet, which is really fun because they are trying to show the horizontal transport due to cilia. And you can see that they are creating, like, they are super organized and they are moving like coherently in one direction, and they are showing that they are transporting something above them. But in the micro scale, we show that if you self-organize like this, and this is if this is cilia, then you have to be like moving randomly actually to transport. And my colleague Arnold then participated in a different work where they put a carpet with only cilia. This cilia, I think, was in the airborne, and they put some tracer particles with the fluorescence, and you can observe how if the cilia is moving randomly, they are transporting actually these tracer particles above the carpet. You see? So what is the future work and the applications that we expect to do with these two papers, or with about the active carpets work? The first one is maybe to study the alga blooms. So at least here in Chile, alga blooms is a really problematic situation since we have a lot of aquaculture things here with salmon and other fishes. And then now we observe that, okay, if micro swimmers like algae is accumulating on surfaces, they can create these transport, vertical transport. And then they can move actually nutrients from let's say here, from different depths in the ocean. And so maybe it's interesting to study how is the real effect of alga accumulation on surfaces. And also now thinking about the hopping effects on active carpets, you can think in a way of self-cleaning devices. Like if you have a really small device in the micro scale, and if you put like these little hairs or like cilia, and you can move them randomly, you know that you will create this self-healing effect or this self-cleaning effect, because microparticles will hop above your carpet, and you will get the surface really like with dirt accumulation. Okay, And also Arnold explored this with another group in United States, where they create this carpet with like artificial cilia, and you can see how they can transport a particle above the carpet. So I hope that you understand the work and you have fun, and thanks for your attention.
Thank you, Francisca. It was really interesting work that you are doing with this. I mean, from the theoretical point of view, it's quite complicated, right? I mean, it's complicated to have all the parameters, and typically you need to do some restrictions to long range or short range, right?
Yes. Mm-hmm.
Okay, so is there any questions from the audience? Well, in the meantime, if nobody wants to ask anything, please, may I say a couple of questions, Francisca, about this classification, let's say, that you made with different active carpets.
Mm-hmm.
In the part of so you have, like, I don't remember, like 4 cases, no? Like the case
Yes.
of the actin, the case of the other that is up and down,
The Vorticella, yes.
the Vorticella, and
Bacteria and cilia.
the dipole, and the
Yes.
other one that I don't know. But in the case of the dipole, this is quite difficult, right? Because as you mentioned, you have this translation translation time, let's say, the time that is related to the self-propelling of the particle, and the other one that is associated to the stress of the fluid, right?
Yeah.
I don't know if you have tried to just imagine that you are able to modify the bacteria to just have the stress part, not the self-propel translation part.
Mm-hmm.
I don't know if there are some results about that. For example, to confine in 2D maybe, and as you know, like this projects that have been done by Dietrich and others in Germany, that they work with this kind of models, but they basically restrict to the 2D movement. So I don't know what.
Yeah.
How could you?
Yeah. I mean, for the dipoles, also the problem that appears is like you can modify maybe bacteria to avoid the run and tumble, because run and tumble time, it's a memory time. Then you have the time that one microswimmer takes to explore the whole surface. And there is also another different time, which is the time related with the self-propelled velocity, because you know that when like a pusher particle moves on the surface, and if you put a single tracer, the tracer performs like a loop, like an open loop, right? And this loop depends, or the tracer movement depends on at which height do you put the tracer. So there is also like a memory time that for the tracer to diffuse, depending if you at which distance do you put it. That's why it's like really, like you can maybe avoid the run and tumble time, like maybe working with this kind of bacteria that doesn't do run and tumble, and they only perform like a direct motion. But still, there are too many things going on with pushers that it makes different to study the active carpets in an analytical way. You can still do it like with numerical simulations, and you can check things, but analytical is very demanding. So it's still something open there.
Yes, of course. And what about this debate that has been done, especially by Leuven, about the how do you say this? The hydrodynamics interaction behind the effects of the collective motion of active particles. Meaning that, imagine that you have chemotactic particles, and there is a debate about if this chemotactic flow field is important to see this motile-induced phase separation, for example. So I don't know if you have some information about it, and if you are you have been working with the other kind of fields. I mean, chemical fields is plus the hydrodynamic fields that you have in your theoretical framework.
No, we haven't explored yet like these chemical flows for like for the Janus particles or things like that. But still, the theoretical framework that we create by studying this problem is quite open, and I think it can be used for different flow fields. Like, it doesn't matter if it's a long range or maybe a short, maybe it's important a long range flow, but it's just a way. You only have to know how is the flow field, and it's also important that we, in the second part, to study the vertical transport, we didn't allow bacteria or particles to self-organize. So there are not hydrodynamic interactions between the microswimmers, between them. That's why we were studying only like really homogeneous distributions on orientations and also on particles in the space.
I see. Okay, so yeah, it's quite difficult, right?
Yeah.
This fixed equation is quite a work.
Yes.
Yeah. I don't know if there is further questions from the attendees, from the panelists. If not, we just thank to Francisca for really interesting talk and really hard work that you made, and hopefully we'll keep in touch to see other works that you have. No? For example, this active Brownian model for contagion.
Yeah, of course,
Interesting.
Okay.
So thank you, Francisca, and we thank to everybody to stay here, and we continue with the poster session. So maybe Eric, can you help me with this? So I can share the videos of the 3 videos that we have in the Drive, Google Drive, perhaps? What do you think? Yeah, Victor?
Yes.
Yeah, so let me see if I can share the video. Just a minute. Because I don't know, the problem will be the audio, right? Share sound.
Let me see, Paco.
Oop. Are you there?
We can hear you.
Do you hear the video?
Yes.
No.
No, I can see, but I can hear you.
Actually, it's in a very bad quality. We see your screen in a very bad quality.
I put here to optimize. Maybe I need to check optimize video clip, stereo high fidelity, advances. So computer audio, right? Now?
No.
No.
I'm sorry, but we can hear.
Photo from second camera, portion of the screen. Stock. So I don't know what to do to check the audio. What do you think? Maybe Norma can talk about the content of the video. I don't know.
Or do you want me to try?
Yes, sure. Go ahead. I stopped to share the screen.
Hi, everybody.
Yeah, I don't know if it's Ubuntu, as Claudia said.
Oh, okay. No, so that means that it is playing correctly, right?
I can hear the video. When you,
Okay, great.
when you did play, I hear the video. So maybe Claudia was correct in the chat when she said that maybe it's Ubuntu. I don't know.
Great. So if you see the display correctly, I mean with good resolution, and hear the video, then I will start, right?
Yes.
Hi, everybody. My name is Sol Maria Hernandez Hernandez, and this occasion in the International Conference on Science and Technology of Complex Fluid, I'm going to present my poster. The name of my poster is Modeling the Dynamics of Tracer Particles in Hydrogels by means of the random walker under confinement. This work is a collaboration between Benemérita Universidad Autónoma de Puebla and Universidad de Guanajuato. The authors of this work are Norma Palmero, Eric Sarmiento, and me. I hope you enjoy my presentation. The abstract of my poster is this. If you want to read it, stop the video and do it. Let me start with the introduction. Scientists in various fields of science is interested in the complex system for many reasons. In particular, we are interested in the dynamics properties of interpenetrating polymers, networks, hydrogels, and world-like micellars. An experimental finding report in this uh, in this journals of matter uh, was mass science distribution. In this paper, they got this graph. Now, we propose a model to study the dynamics of tracer particles in hydrogel. The model consists of two things. One of them is two-dimensional random walks under confining conditions, and another thing is the ensemble of the walkers in the boxes of the different signs. We designed three different confinements. We will call the first one weight. Weight methodology consists if the wall reaches the wall or it's gone out of the box, he waits. He waits on the wall or near of the wall from the next random step that takes it away from the wall. Looks like this. The second methodology consists when the walker is close to the wall and next random step takes out of the box. Then the walk reaches the wall and rebounds in the opposite direction, covering the distance indicated for the random state. We will call rebound. Looks like this. The last methodology is reflect. He avoids touch the wall when the walker is close to the wall, and the next random step takes out of the box, then the walker go to the opposite direction, covering the distance indicated for the random step. Looks like this. Second part of the model is an ensemble of the walkers in the boxes of the different size. We basically imitate hydrogel networks by design boxes of the different size. We use a Gaussian generator with a given mean and standard deviation. This graph represents two area distribution with the same mean and different standard deviation. When the standard deviation is small, the area of the boxes can be fit a Gaussian curve. It's not the same when the standard deviation is large. The main results of this work is in these graphs. We obtain the mean square displacement for different confinement, and we can see the difference is in this region. And we obtain the mean square displacement for the different standard deviation, and we can see this. Well, finally, if you have a question, write me. This is my email and my Twitter. Thank you very much.
Okay. Thank you, Eric. So we'll continue to the other one, right? There are 3 videos, right?
Okay, just give me a minute. Full screen.
Hello everyone, welcome. Hello everyone, welcome to the video presentation of my poster entitled Single Trajectory Analysis of Droplets Produced by Spontaneous Emulsification at Water Glycerol Dodecane SPN 80 Interface. Although in this video I will only show some fragments of my poster, the complete design is inspired in a type of a research notebook, of course in my own particular style. And now, let's go to my work description. Spontaneous emulsification consists of placing two immiscible liquids in contact that will start an emulsification process without supplying external energy to the system. In this work, spontaneous emulsification at the water glycerol dodecane SPN 80 system was analyzed. Previous work by my laboratory partners found that droplets produced by spontaneous emulsification at a curved interface assemble into a two-dimensional colloidal crystal. In addition, the droplet size depends on the initial concentration of surfactant, and it is also a function of time. The droplets move over the interface and then sediment, forming the aforementioned colloidal crystal. In my case, the interface consists of a concave meniscus, as I will explain next. As far as my work is concerned, we have distinguished 3 stages in the colloidal crystal formation. Nucleation. Some droplets are located at the lowest point of the meniscus. Intermediate stage. Crystal grows mainly by the new droplets added. And final stage. The crystal grows mainly by the contribution of the droplets growth present, and only few droplets are added. It is important to say that in this work we present a brief description of the droplet's motion on their way to the colloidal crystal at the final formation stage. The experimental setup consists of a rectangular stainless steel cell with two cylindrical concentric holes. At the holes are perpendicular to the plane of the cell, and water mixed with 1% glycerol is placed in the upper hole, while the decant mixed with 2% span was placed in the lower hole. The emulsification process happens at the interface, and as I had said, in this configuration the interface is a concave meniscus, which in the observation region is considered as a plane, because the curvature radius of the meniscus is much larger than the cell radius. We observe the interface using an inverted microscope, and due to the surfactant characteristics, we observe water glycerol droplets in the decant. It should be noted that the entering meniscus is not observed in each experiment, as priority is given to the droplets-free region and the crystal boundary to observe the addition of new droplets in detail. For example, it is a typical observation at intermediate stage of crystal formation. Furthermore, the presence of glycerol slows the droplet movement, allowing that the observation region trajectories of greater length to be maintained than those obtained without glycerol. Since the droplets on the crystal continue to grow, the recorded times are about 120 seconds, time in which it can be considered that the crystal boundary is approximately in the same place. The observation regions are distinguished with respect to the colloidal crystal boundary, distant, intermediate, and nearby droplets. As shown in the figure, the droplet's motion is a Brownian motion with drift. The velocity decreases as the droplets approach to the crystal boundary, where the droplets behave similarly to approach a wall. It is important to mention that each trajectory is unique, probably due to the interface irregularities. For this reason, each trajectory was analyzed individually in its x and y coordinates, sectioning it where there was a change of direction and therefore velocity. For each trajectory fragment, the velocity mean square displacement as well as jump distribution of each of the droplets were calculated. In order to verify the obtained results, we subtracted the velocity of each trajectory fragment to recover only the droplet's Brownian behavior, and we sum the contribution of the trajectories in each region. The results show that the density probability histograms are equivalent for each region at different times in the same experiments. So the droplets behave similarly in equivalent regions at different times. And that's all. Thank you for your attention.
Paco, do you want me to
Yes.
play the third one?
Yes, sorry, yes, no. So we finish with this, and we will ask if somebody wants to describe his poster, if you agree.
That's so.
And then we continue with the questions if there are some questions about the videos, and that's it, no?
Yeah.
Perfect.
That's it. Let me continue with this one.
My name is Norma Palmero. I am a PhD student in the University of Guanajuato. I will present you my work entitled Hydrodynamic Correlation of Trapped Particles in Optical Tweezers. The main motivation of this work is to understand the hydrodynamic interaction in complex fluids, as those are relevant in several branches of science. Phenomena such as hydrodynamic synchronization in either biological system, sperm, cilia, flagella, active collateral rise, and the dynamic of macrostreams can be explained instead of hydrodynamic coupling. In this work, we report experimental results corresponding to a direct measurement of hydrodynamic interaction of two microscopic Brownian particles separated a given distance and trapped in optical tweezers. The experimental setup used in the study is divided in two parts. In the first part, the lasers, some optical component, and the spatial light modulator are used to produce several optical traps. In the second section of the setup, optical trapping is captured by a camera and viewed at the computer. In this video, I show you two particles of 2 microns in diameter trap-independent optical traps in separator 3 microns. Hydrodynamic interaction can be analyzed through the cross-correlation fusion of displacement. This fusion describes how the motion of the particle is affected by the motion of these neighbors. The analytical correlation fusion of two particles trapped in optical tweezers depends on the separation distance between the particle, the optical thicknesses, and the friction coefficients. Here, I show you an example of the previous equation corresponding to the theoretical experimental result presented by the other authors. Using the position of the two particles trapped by video microscopy, we compute the cross-correlation fusion of displacement for different laser powers. Here, I show you all the experimental results correspond to the average of 20 experiments at 500 frames per second. For example, at the same distance between the particles, we can know that the minimum for this fusion is deeper if the laser power is low. In this case, the particles have more mobility, and their motion affects the dynamic of the other particle. Similar occurs when the distance between the particles is shorter. Here, you can see the experimental result compared with theoretical model, finding the good agreement between them. For this case, the distance between the particles is the same, but the optical thicknesses are different. Thanks to the organized committee for giving me the opportunity to show you these results, and I will be willing to discuss or work with anyone interested by Twitter, or you can write me to personal email. Thank you.
Nice. So maybe if there is any interesting in talk about their work that are presented by poster, maybe you can raise your hand now, and we will open the microphone for you.
Even we can give panelists permission for the attendees so they can share, for example, the poster just briefly.
Exactly. Exactly. Not only open the mic, right?
Mm-hmm.
So please, is there anybody that wants to talk about the poster? No? Okay. So, Claudia. Thank you, Claudia. So, Eric, can you please allow Claudia to talk?
Claudia, you want me to give you permission for panelists so you can share something?
Hello, can you hear me?
Yes.
Well, yes, well, I can just quickly share if you give me permission my screen.
Okay, give me just 1 minute. You will receive a message. Okay, there you go. So you are not a panelist. You can open your microphone, share your screen, wherever you want.
Yeah. Hi everybody. I just want to invite you to go and check my poster. The title of my poster is "On the Reforming Effect of Solid Inclusions in Colloidal Gels," and I talk about how if we have a colloidal gel and we replace some of the colloidal particles by solid inclusions, we can stiffen the material. And it turns out that the size of these inclusions that we use to replace the particles, the gel particles, it matters in how much the material is stiffened. Well, go and check it, and you can ask me questions about it. So that's all. It's also a continuation of what Anina, going further into what Anina talked with you. Yeah.
Okay, thank you, Claudia. I don't know if there is anybody that wants to talk about their poster. It seems that, okay, it's Marcelo Padron. So can you please give permissions, Eric?
now can speak, but just the same. You, Marcelo, you want me to give you permission of sharing screen?
Can you hear me?
Yes.
I can share the screen.
Okay, just give me 1 minute. So you now have the permissions.
Can you see? Okay. So my work is about unequal radius modified Gauss-Chapman theory for the electrical double layer in several geometries. These geometries are planar, cylindrical, and spherical. In the literature, you can find that if you use this theory, you can see the phenomena of charge reversal and charge amplification. So we demonstrate in the work that it is possible if you use a low value of potential initial that was our parameter of star. And also we present for first time information about cylindrical geometry, and we conclude that charge reversal and charge amplification also can see while you use values of potential low. And we compare the three geometries to search a relation between the geometry and the phenomena, but no present any relation between the geometry and the phenomenon. Okay, thank you, Marcelo. I don't know if somebody else wants to talk about their poster. At 1, at 2. No? Isela. Okay, Isela. So the same protocol, right, Eric?
Yes, working on it. Working on it. Okay, there you go.
All right, so let me see here. I can share my screen. So, well, my poster is about the effect of mask and vaccination in the contagion dynamics by using active Brownian particles. It was developed by myself and the Dr. Alarcon. And in this project, we were interested in describing the dynamics of contagion of a disease or an infection, and we did it by describing the individuals with Brownian active particles, and we base our work in the Lagrange equation that describes the movement of Brownian particles, and we have an overdamped simplified expression of the Lagrange equation, and is the one that we have expressed here. And we had a potential interaction in between the individuals that is repulsive. So then we had a set of rules of the interaction between the individuals. For example, we had individuals that were categorized as infected, susceptible, and protected. So we have different parameters that we work, and we had a box that had a total of 300 individuals, and we start with one infected individual that will begin in the corner of our box and will be introduced to inside where all the particles are contained. And the other parameters that we vary were the force related to the mobility, which we call FA, and we varied from 40, 60, and 80 with different densities of individuals ranging from 0.1 and 0.75 and 0.05. And we had different rates of protected individuals that ranging from 0 to compare it with the rest of them from 0.1. Well, 1/10 of the population and 1/4 of the population and half of the population. And we have that this time, the time that is showing my plots is normalized with the stocks time. And for example, for the results of the first step parameters, we can see that there is indeed a relationship between the way that the rate that infected and the protected, and this interaction or this effect is more evident when we have at least 50% of the population protected. In the other cases, is less apparent, and even we have a case where they contagion even faster. And for the other results, as we have graded velocities, the effect for the 10% of the protected individuals is less evident. And here, the 10% and the 25%, they were pretty much the same. And it was for N80. And lastly, well, it's kind of the same. And in this case, for the lower the density where it's the lowest, it was pretty much the same with the 10% of the individuals protected. There was really not that much change. What it was pretty much the same for all the cases is that the individuals, the rate of contagion was less. It had a greater change when we have 50% of the individuals protected, which means that, well, which also points out that it's really important that we stick to the protocols and safety measures to avoid or reduce the risk of getting infected and maybe flatten out the curve. Like, that's what we want. And well, that's all by my side. Do you have any questions?
Thank you, Sela. Eric, well, I don't know. Sorry, not Eric. I mean, all the attendees, I don't know if there are some other poster presenters that wants to talk about their work. What? One, two, okay, Hector. Eric, can you please give permissions to Hector?
Yes. Should be joining now as a panelist. There you go.
Hi.
Okay, go ahead, Hector. Yeah.
Do you hear me?
Yes.
Okay. I need to share my screen. Okay. Yes. I start with my presentation. Okay. First of all, I need to say because I need to speak about the analysis of the implementation of the VIXED model. First, that's all. I speak about the internal parameters of the VIXED model, like density of particles, the noise, the coating radius, like what is the distance between the particles can say no can watch other particles. First, I speak about the importance of the density in my system. Suppose that you are in the Zócalo of Mexico, and all the people need to go in a specific place, like a bar, like a disco. The question is, which is the easiest way to know where to go, with a big density or a low density? With the model, the BigSec model, the result tells us that if you increase the density in your system, our particles will be aligned faster than a low density. It's an interesting result, and result obviously maybe, but it's an applied result from the BigSec model.
Hector, excuse me, Hector, you are not showing the presentation mode.
No?
No.
Okay. So sorry. Only not. Okay, then let me check. Now?
No, but
No?
maybe you can continue like that, and
or someone can presentation my poster, maybe.
I don't know. No, but you can continue like that. I mean, just in the, I don't know, in the other version, in the other way of seeing the PowerPoint.
Okay.
So share your screen and then put the presentation mode, maybe.
But I don't know if you can
I have the poster.
my...
Yeah, I have the
okay.
poster.
Okay. You check my poster. Okay.
Yeah.
Well,
analysis.
for the first part, I speak about the implementation. I speak about the density, the importance of the density in my system. If you increase the density, your particles will align better than a low density. It's a result, obviously maybe a result, but it's an interesting result because I can check other internal parameter like the noise. The noise, maybe you can see like the people speak other language. If you stay and you need to go someplace, if you say, no, I don't know, Juan, where are we going? Juan speaking Chinese or in Japanese, and I don't know. It's difficult to understanding the both. It's in the Vixen model when the noise in the system is increased, so you increase the language that the people speak, maybe. We can analyze the system behavior like this. The noise increase, the particles like, I don't remember how do you say panal de abejas, but it's like this. In the order in the system, when I increase the noise, obviously the order decrease. Then the next part, I speak the simulation in the Mesosim lab. We worked. We have, for the moment, 3 types of collisions. That the bones collision, that is when the particle crash with the obstacle. The particle boings, like rebota, in the half P grades. The elastic condition is like the law of the Snell. the bacteria condition, the particles go on the face of my obstacle. But no, this type of study are in standby because we are focused on the particles attrapment, like maybe tweezers opticals. In the BSEC model, for the moment, we are trying to describe this type of system. When I put a circle like a trap, and my particles are trapping, I don't know if I can speak this part. The particles trapping in my trap, and I check the order because when I give other power in my trap, so it's sorry, it's difficult to me speak or presentation something, but it's to finish this this presentation when I variance the power of my trap, I have these different results, and obviously the order in my system is different. The interesting result of this simulation are that if I use a bigger trap with any noise, I trap my particles. It's interesting because I don't import what is the power of my trap, but I will attrapting particles. And sorry, my English is like before president, but I will try to learn more English language. That's all.
Opa, ¿dónde está? Okay. Thank you, Hector. I don't know if there are somebody else that wants to present their poster.
Karen wants to present her poster.
Yeah, I have a problems with the Zoom. Eddie, can you take the organization of this thing? Because I think I have a problem with the Zoom right now.
Okay, don't worry. Karen, you should be now promoted to panelist.
Hello.
Hi, Karen. We can hear you correctly. Whatever you want, you can share your screen.
Hello, can you hear me?
Karen?
Hello.
Karen? It seems that we lost Karen. Okay, so Karen, we hear a lot of noise when you open your microphone. So I'm not sure if you are in a very noisy environment or there is a problem with your hardware. Let me try again.
Hello, can you hear me?
Yeah. Yes, we can hear you, Karen, but it's quite loud, the noise of the background. So it's even kind of disturbing. Paco, I don't know if you want to maybe change the role again, or do you want to try again with Karen?
Do you hear me? Can you hear me? Hello?
Yes, yes.
Yes, well, I don't know what is the problem with Karen. What do you suggest?
She just joined from another device.
Ah, perfect.
There you go.
Now we can hear you perfectly, Karen, I think.
Great.
Okay, thanks. One moment, please.
Yes.
Okay,
Perfect. When you're ready.
okay, thanks. Well, good morning, everyone. This time I present the work Mesoscopic Simulation of a Virus by Dissipated Particle Dynamics. Currently, the 2019 pandemic was responsible for numerous deaths around the world, thus representing a danger to public health. The main mode of spread of a virus like SARS-CoV-2 is the short-range transmission of infection saliva droplets. And studying the hydrodynamic interaction of a virus will help us to understand its transmission through solvent. This transmission is related to temperature and that spread of the viruses changes with the season of the year. And because use the DPT model, we can take the temperature as a variable. Then biological complex system need numerical simulation to understand from a meso and microscopical point of view different properties of viruses. And thus the objective of this work is to study the hydrodynamic transport of viruses by implementing the algorithm DPT. Well, now in the same numerical framework, we've used the icosahedral geometry of the virus since it has been studied as a model type of equilibrium structure of viruses. The solvent and the virus are represented by GPT particles and the rigid body models of the viral capsid which different radius contacted with lamps. In this work to study the dilute regime, we have carried out systematic simulation of a single virus immersed in water with period boundary conditions, where the diffusion of the virus capsid increased with the temperature, but when the capsid size was increased, the diffusion decreased. So dilute regime performed is more detailed model of SARS-CoV-2 virus. The first results found are related to the effect of the size of the SARS-CoV-2 model and the implementation of protein S with the hydrophobic S2S domain. Okay, the results show that the implementation of the S protein in the SARS-CoV-2 model affect the mean square displacement at short and intermediate times. However, at line times, there were not significant changes. Well, the study of the fusion and an important role to understand the transport mechanism of a virus. This will be associated with outbreaks of infection through the temperature of the medium. Thus, we concluded that our numerical approach is consistent to experiment results obtained by the book and Altman. Thank you very much for your attention.
Thank you, Karen. I don't know if there are some other panelists that wants to share your work.
Just raise his hand. Let me promote
Yes.
to panelist.
Thank you.
There you go. Arturo, you can share your screen whenever you want.
There is another one, Jesus Cepeda, okay? But after Arturo, I think, no?
Yes, Arturo.
Can you hear me?
Yes,
yes,
correctly.
Okay, I'm going to present you my project that is related population contagion dynamics using active Brownian particles and probabilities of contagion as a model in computer simulation. This project was developed about Francisco Larcon and me, Arturo Jimenez. So the motivation of this project is to and was based on the work of Francisco Guzman. So it is to have another alternative of classic contagion models. So in this model, we propose to use active Brownian particles. So we have n particles in a square box of size L with periodic boundary conditions with a self-propulsion force that we name FA, and for each particle is associated to the self-propelled velocity. So we start with one infected in the simulation. So and there is the contagion now. I used first a probability of contagion like 1, having only in the box that one contagion condition, no, that if one infected particle was close to a susceptible particle and were less than a distance RC, that is the distance of cutoff radius, and the susceptible particle will be infected. This is one of the condition that I use, but we use another condition, that is to associate the susceptibility of the particle to a probability. So such the distance of the infected particle and the susceptible particle were less than the cutoff radius. We set a random number, and if this number is less than the probability of contagion, and this will be affected. So this is in general the model that we use. And in the right side, we have the results about the simulation. First, in A, B, and C, we have the results where we have the one probability. So we conclude this probability that the contagion dynamics will increase if we increase the the self-proposed velocity. So in D, E, and F, and we can see that I use respectively 0.75 the probability, 0.5 and 0.25 probability. This is how to discuss because we can see that every plot is similar, and we can notice there's a very little difference, and it's because we have a lot of simulations in our time step, and so every time we set the random number, we set that in every time step, so it's a lot of tests. Then there is the reason we can see that in the condition will be similar like the other, how can I say, and that we have a less difference about the one probability. So I don't know, I'm sorry for my English if I were clear about my presentation. So I thank you for your attention.
Thank you, Arturo. So, Jesus Cepeda, are you going to present your work? Please raise your hand again to say yes. Okay, Eric, can you please move Jesus to panelist?
In process. Jesus, you can now share your screen.
Hello, I'm Jesus Cepeda, and I am going to present my poster titled Waiting Time Dependent Non-Equilibrium Phase Diagrams of Simple Glass and Gels Forming Liquids and its Morphology. I'm currently having some problems with my internet connection. I hope I can fulfill this talk. So the main motivation of this work is basically to prove that we can use this theory, the non-equilibrium self-consistent generalized Langevin equation theory, to obtain diagrams of systems in which the response times of the transformation is relevant for the description of the systems. Such happens with like borosilicate glasses, as it's displaying this kind of diagrams where the waiting time of the system is part of the description. And for example, another interesting system is the Laponite clay systems, in which one can expect gel and glass-like phases, depending on the concentration of polymer and Laponite. So what we do is we use this sort of theory, which, let me make a quick sum. We make use of this theory, which basically consists in two main equations: an equation for the evolution of the density, which is basically a fixed law, in which the only difference comes within the mobility of the system. It's a mobility which it's actually calculated or obtained through the state of the system, and an equation for the covariance of the mean density, which is basically an equation for the evolution structure factor, These two equations are then coupled through the mobility, which is obtained through a set of equations in an MCTO-like scheme, which are coupled through the intermediate scattering function, its self part, and a memory kernel. So one can obtain the mobility within this set of equations, just as shown in this equation. And we use this formalism in order to derive the evolution of the system. So let us, in this work, we make the example through a hard sphere square well interacting fluid system, which is characterized through this two particles interaction potential. So once we select the systems, all we have to do in order to solve the equations of the system is to propose this sort of thermodynamical property, which we can input as the main external factor to the set of equations that I just showed above. So what we obtain when we do this, we have to propose actually a protocol for the system evolution. So what we do is we start from a hard sphere limit, which in this system is at infinite temperature, and we quench the system instantaneously and isochorically. So we can monitor the behavior of, for example, the mobility of the system. And so we can obtain this sort of diagrams in which we can visualize the mobility of the system accordingly to the phase states of the system and the waiting time or the preparation of time of the system. So once we solve this sort of instantaneous quenches, we do it through a set of multiple final states of the system, so we can obtain this sort of cuts through time and depending on what are you interested in. You can make cuts in, for example, the density and the temperature, or or at constant waiting time, or for example at constant final temperature and along the phi and along the density and the time planes. So you can obtain this, and all we have to do now is just to make a proposal in order to obtain these sort of diagrams that I have shown you here, in order to understand the transition towards this sort of glass and gel-like states of the system. Within this kind of theories, the usual proposal comes along when the mobility of the system reaches zero, yet it is not a practical application to have such threshold for the mobility. So what we actually do is we just set the threshold of the mobility to in a more practical sense, in the same sense that, for example, for glass systems, for colloidal glass systems, the viscosity, you can set the threshold of the viscosity as 5 orders of magnitude greater than from a dilute system, in order to how do you say it? in order to in order to take into account the glass transition of such colloid system. So right now, I'm just going to show you the resulting phase diagrams when we make the analysis for the mobility, and we are going to ignore for now these images that are going to be there, and I'm going to explain them soon. But we can follow the mobility through a color scale map, just as the one we use here. And we set the threshold through the isomobility line at which is 5 order of magnitude lower than the typical dilute system. So we start from this sort of mobility across the phase diagram, and when we quench the system in consecutive times, for example, at t=1, we can actually follow the mobility lines, isomobility lines through the landscapes of the thermodynamical properties of the system. So for example, in this image, we show the isomobility line for 10 to the minus 1, 0.1, and we take the threshold and we set this thick gray line as the equilibrium line that we expect for such isomobility. So what we expect is that this isomobility line that we are following at a certain time eventually reaches this fixed solid gray line. And right here, we are still not seeing any isomobility line of 10 to the minus 5 because the system states are actually pretty movable. So we actually have to wait a little more in order to follow such line. And this line is followed through the solid black line. And this is the line that actually we set in order to in order to take into account the glass transition or the arrest transition of the system states. So this system actually presents an spinodal line, which is a sort of like the mixing line of fluid states. It mixes into a gas and liquid states. The system mixes into gas and liquid, and such line it actually coincides with the isomobility line that we were following through the dotted black line. So in this region, we are actually inside the spinodal line, and what we follow is the dynamical arrest transition set at the threshold value of B of the mobility equals to 10 to the minus 5. And when the evolution of the system when the time evolution of the system increases, what we expect is for these isomobility lines to eventually coincide to the spinodal line. So this dynamical arrest line that we are following actually moves and eventually reaches also this black dotted line. And in addition to these waiting time phase diagrams that we are showing here, we can actually also follow the morphology of the system through the evolution of density fluctuations. And this is what we are showing right here in these inside boxes for state-centered final states. So what we are actually seeing here is the evolution of the density fluctuation across the waiting time phase diagrams predicted by the theory. So for example, for states outside of the spinodal line, an equilibration process occurs, which is simply the homogenization of the system density. While, for example, for states inside the spinodal line, such the mixing translates into large structural heterogeneities within the density. These are the sort of prediction that we are now calculating and trying to compare with simulation data. So this is like it's a perspective of this work to eventually compare this sort of density fluctuations results with morphology found within simulations. So we can actually state some general tendencies across the waiting time phase diagrams and these morphologies. And I invite you to look up for my poster and ask me some questions later. And that's it for now.
Thank you, Jesus. Okay, so is there anybody that wants to present their poster? Or if not, we can finish with Jesus. And with this, we finish the morning sessions. We will restart after lunch at 4 p.m. I don't know if there is another announcement, Eric.
I don't have any announcements, Paco. Thank you.
We will try to open this gather session for in order to have rooms for talk individually or with the little groups, right? So yeah, so we'll keep informed about that.
We can try maybe later tomorrow.
Mm-hmm. Mm-hmm. Okay. So see you later. Thank you very much to all for attend and for participate, especially for let me thank to the invited speakers, to the contributed speakers, and especially for the participants for the posters that even though that they don't send the video, they have this effort to present their results, and of course to the people that send the video also. Thank you. Thank you, Eric.
See you later. Bye.